Hi students, am I audible? Hello. Hi everyone, am I audible to you? Hi kids, am I audible to you? Hello. Hi everyone, I hope I'm perfectly audible and visible to you all. Hi, good evening everyone. Do let me know in the chat section, am I perfectly audible and visible to you all? Am I perfectly audible and visible to you all? Okay, good evening, good evening, good evening. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma and I have a big news for all of you. I hope you all have seen this particular video. If not, then please go to this link. Right students, this video, do watch this video and you have to hit a like button here and students, remember one thing, right? See, this is going to be something very amazing for all of you. Trust me, this is going to be something very amazing for all of you. So make sure you watch this video, make sure you like this video and just wait for a few days and there is a biggest surprise for all of you. There is a biggest e-revolution for all of you, right? Okay, so quickly hit the like button without delay. Quickly hit the like button and do watch this video. We'll tell you what is the surprise for you and trust me, it's going to be very amazing for the NEET 2025 aspirants as well as for the NEET 2026 and 2027 aspirants. Okay. Okay, so tell me, how are you all? How's your preparation going? What are the things that are bothering you? And what about the biology? Tell me everything about that. Then I'll start the session. Yes, Rohit. Then definitely I'll start the session. First, answer my questions and hit the like button. Do subscribe to our channel and share the session link with your friends. Invite your friends for this wonderful lecture. Because within 4 hours, we are going to finish one more chapter, that is excretory product and its elimination. So students, as promised, right, as promised that we the complete team Avengers, we have ensured you that we are going to complete your syllabus here, right, and we are doing it. But if you people will not support us, if you people will not watch the lectures, if you people will not attend the classes consistently, then tell me, how are we going to stay motivated? Of course no, how are we going to stay motivated? Even we will feel sad. We are also going to feel sad. Okay, so when it comes to your biology preparation, you know that yes, you all know this thing that in biology, physiology, be it human physiology, plant physiology, they are very important. Cell biology, very important. Your ecology, very important. Your genetics, very important. The complete reproduction unit, very important units they are, right? So here in this particular series, in the NEET Endgame, I have started with the most important part and I will proceed accordingly. Okay, I will proceed accordingly. But on Unacademy NEET English channel, you can watch Game of NEET 1, Game of NEET 2.0. You can watch our bounce back series. You will find everything on this channel, right? You are going to find everything on this channel. And these one shots that we are taking these days, they are also amazing because here we are not compromising any content. Trust me, we cannot, at least, right, the team Avengers, we cannot take the one shots for 1 hour or 2 hours because we know that when we have to deliver the information, it is going to take 3 to 4 hours, right? It needs 3 to 4 hours. Okay, so be consistent, guys. Right, be consistent, guys. Let's finish the human physiology, then let's move to the genetics, then let's move to the ecology, then let's move to the plant physiology. So be consistent, right, and just keep on studying, and trust me, if you work hard, you're definitely going to get a good result. Trust me on that. You're definitely going to get good result. Okay, chali. So let's start the excretory product and its elimination, but make sure you have liked that video because something amazing is waiting for you, and you know that that is the biggest event ever, and that is ARAM 2025. I hope you all have seen ARAM 2024, and this time it's ARAM 2025, and you know now every year we learn something new, right? Every year we learn something new, we get better. Same is going to happen here. Same is going to happen here. This event is going to be very amazing, right? There will be surprises, there will be mega rewards, fun will definitely be there. So you have to be live on YouTube on 1st March 7 p.m. to 9 p.m. Okay, we'll share the details, we'll tell you about it, right? Today let's So this is all about today, but yes, stay tuned to our channel, stay tuned to the videos, right? You'll get the details as well. Okay, you will get the details as well. So let's start. And dream crash course bache, it is also going very well. You can ask the students, right? You can ask the students. For biology, their physiology and genetics is going on, right? It is going on parallelly. So our target is to finish the most important chapters, then let's move to the most important topics. That is the target here, right? And we set the realistic targets. So guys, make sure you join the dream crash course batch. In the description box of this particular video, you will get the link. Click on that link, use the coupon code Ambika10, and you will be the part of our batch. Okay, so the first test we are going to conduct on 23rd of Feb, so that you will get some time for the preparation. So without any delay, be the part. Okay, without any delay, be the part. Okay, so let's start the chapter now. Let's start the discussion. Fine, but do not forget to like the video. So excretory product and its elimination. So we will start first with the word excretion. So you tell me, what do you understand with the word excretion, and how is it different from defecation? What do you understand with the word excretion, and how is it different from the word defecation? Yes, please. Yes, please. Yes, please. Tell me quickly. Tell me quickly. Let's have discussion. I don't like such boring sessions, right? Where it's too, I'm just saying, I'm just writing, and students are like, okay, let's chit chat. Hi Ria, how are you? Hi Angel Priya, how are you? I really don't like such kind of sessions, right? So let's have interaction, guys. Let's have interaction. Okay, let's have interaction. So what is excretion and how is it different from defecation? That's my first question. What is excretion and how is it different from defecation? That's my first question. Please try to answer that. Please try to answer that. Yes, please. Please try to answer that. What is excretion? Removal of nitrogenous waste. Okay. Removal of waste. Yeah, removal of nitrogenous waste. And what is defecation? Then what is defecation? Removal of undigested waste from the body is known as what? It is known as the defecation. Yes, yes, people, you are right. Now we have to understand it in a better way. Just a minute. Okay. Today's target is clear. Chaliye. So as you all said, right, we'll start with basics first. So as you all said, yes, excretion is the removal. It is the removal of nitrogenous waste. Without any doubt. You know that when you talk about your body, let's say proteins, they have the nitrogen in them. Nucleic acids, they have the nitrogen in them. So basically when there is that, animals, they accumulate this nitrogenous waste. Not just the nitrogenous waste, other things are also there, right? Majorly we say this, that when we talk about the excretion, we are saying there is the removal of waste, and particularly metabolic waste, particularly this nitrogenous waste. That's what we say. This is what excretion is. We'll further understand it. Now in a very simple way, I'm trying to explain the same things. When it is defecation, right, so defecation is what? It is the removal of undigested material from our body, right? It is the removal of what? Undigested material of our body. This defecation is your excreta, right? Your excreta, your waste, your undigested food, or you can say that potty, excreta, undigested food. That is the defecation. That is the defecation. Sometimes students, they don't understand the meaning of defecation. That's why I'm explaining it like this. So excretion is the removal of nitrogenous waste, and when I'm saying defecation, it is the removal of undigested material from our body, right? Removal of undigested material from our body. That is what defecation is. It's simple. When it is the removal of nitrogenous waste or metabolic waste, right? Nitrogenous waste, or you can say metabolic waste, means metabolism. The word here is metabolism. Metabolism means the chemical reactions which are going on in our body, right? What is the word metabolism here? It is the chemical reactions that are going on in our body, right, bache? And you know that we all are alive because of these chemical reactions only, right? We are living because of these chemical reactions only, right? This metabolism is very important. Being living and this metabolism, these two things, they are synonyms, right? Being living and metabolism, these two things are what? They are synonyms, right? If we are living, means chemical reactions are going on. right? Right? Means chemical reactions are going on in our body. And when you talk about these chemical reactions, right? When you talk about these chemical reactions, okay, so catabolism, anabolism, both the things are there. There is the build-up, right? There is the build-up and there is the breakdown. There is the build-up and there is the breakdown. Both the things are going on in our body. So if we are talking about the metabolism, means that reactions due to which we are alive. So metabolic waste is the result of that reaction. Means something has to enter in our metabolism, then the waste, that removal of that waste is the excretion. But when the food is undigested, right, means it was never the part of our system, right? Right, it is not the part of our metabolism. Are you getting my point? So that is the difference in these two terms. Okay, that is the difference in these two terms. Done? Done? So basically what's going to happen? The animals, they produce the certain nitrogenous compound or materials which are of no use in our body. So this is the nitrogenous waste material. Okay? Okay? And why is it important? To regulate the composition of the body fluids. Right? Why this excretion is important? Because to regulate the right, to regulate what? To regulate the composition of body fluids. Fine, that's what we have. This is what excretion is. Understood? This is what excretion is. And of course, for the excretion, what is required? Excretory organs are required. What is required? Excretory organs are required. And we have to understand that what exactly the organisms are going to excrete. But first of all, before going forward, right? Let's talk about the, I'll start with the basics first, then I'll go to that types of nitrogenous waste. Okay, chali. So now tell me, what is homeostasis? Anybody? What is homeostasis? What do you understand by this word? Homeostasis. Anybody here in the class? What do you understand with the word homeostasis? Quick. Yes, please. What do you understand with the word homeostasis? Yes, you are right. Kalyani is right. Afreen is right. Very good, beta. To maintain the constant, right, conditions, right, the maintenance of the constant condition, or you can say that it is defined as maintenance of steady state, right? So in our case, you know it very well. Let's take the example of human beings. Right? In our case, you know that our internal conditions, they are constant. They are not changing with the surrounding. Right? We are warm-blooded animals. Right? We are endotherms. We are warm-blooded animals. Right? We are homeotherms. We have the constant internal conditions. Our constant internal conditions, they are not changing with our internal conditions are not changing with the surrounding. Right? They are not changing with the surrounding. And how is it possible? It is possible because of osmoregulation. It is possible because of thermoregulation. These are the things. And yes, these things are energetically very expensive as well. Okay, these things are energetically very expensive as well. That's a fact too. Okay, that's a fact too. So homeostasis is the word. Homeostasis is the word. So it is the maintenance of steady state. Right, bache, the constant internal condition. And to maintain that constant internal condition, right, there are mechanisms which are going on there in our body. Right, metabolic processes are there. Right, so as I said, the salt and water regulation, the thermoregulation, all that things, right, they maintain the constant conditions in our body. Okay, bache, then you tell me, what is the meaning of osmoregulation? What is the meaning of osmoregulation? Yes. What is the meaning of osmoregulation? Yes, please, Stella. What is osmoregulation? Like, this is how we define now that it is the maintenance of salt and water balance. Yes or no? It is the maintenance of salt and water. Yeah, to have salt and water balance in our body. Alright? To have salt and water balance in our body. This is the osmoregulation, and this is also something important. This is also something important. Right, bache? Yes, you are right. The maintenance of salt and water balance in our body. Okay, bache? Okay. So this is the osmoregulation. So basically what we have to do, all that things are required. Right? This osmoregulation is very important as well for the conditions like homeostasis, and that's not mandatory that osmoregulation is going on in our body, then there will be the maintenance of steady state in all the organisms. That's not that's a very different discussion. Here we have to talk about the cold-blooded, warm-blooded. Let's not enter into that. Let's stick to this topic only. So I'm just explaining few terms here. I'm telling you about what homeostasis is, how is it possible when there is a proper osmoregulation and thermoregulation in the body. Overall osmoregulation, that is also important because there is the maintenance of salt and water balance, right? Like if I take the example of the amoeba, you know, it's a unicellular eukaryote, right? Amoeba, you know, unicellular eukaryotic organism it is, right? Having that irregular shape. It lives in the fresh water. So it is also having the contractile vacuole, if you remember, right? It is also having what? It is also having the contractile vacuole. So because it lives in salt and water, it lives in fresh water. So just to have, because metabolism is going on here, surrounding is also there, metabolism is also going on. So just to maintain that salt and water balance, the contractile vacuoles are present here, right? But the contractile vacuoles are present here. Are you getting my point? Are you getting my point? So osmoregulation is something very important. So some organisms, they are osmoregulators, some are osmoconformers, right? We'll explain it as well. Now let's move to this NCERT part, and from here, because these are the short one-shots, so we will mainly stick to the NCERT and accordingly we will discuss. So they are saying that animals, they accumulate ammonia, right? Urea, focus on this, they all are nitrogenous waste, and uric acid, nitrogenous waste, then CO2, even the water and ions like sodium, potassium, chloride, phosphate, sulfate, etc. Yes, they do accumulate. When you talk about the animals, yes students, the animals, they accumulate ammonia, urea, uric acid, carbon dioxide, other ions as well, right bache? Right bache? And this is due to, how is it possible? Metabolism and indigestion can be there. So these substances, they need to be removed from our body, right? Either totally or partially. Matlab their removal is important because it's a waste. It's a waste. Like if you talk about the lungs, right? If you talk about the lungs, right? See, you know that lungs are the respiratory organ. They play the role in exhalation. So when we exhale, the CO2 goes out. The CO2 goes out. So carbon dioxide is also the metabolic waste. That carbon dioxide is also the metabolic waste. So in this chapter, this is what we are going to learn, right? We will talk about the removal of these waste, particularly nitrogenous waste. So when you are talking about the nitrogenous waste, so ammonia, urea, uric acid, they are the major forms. They are not the only forms, let me tell you. They are the major forms, right? Again, I'm saying they are not the only forms, but they are the major forms. Ammonia, urea, and uric acid. So removal of ammonia is ammonotalism. It is ammonotalism, and that organism will be ammonotalic. That organism will be ammonotalic. Urea, removal of urea is ureotalism. It is ureotalism, and uric acid is uricotalism. So while answering the question in paper, read these two words carefully, right? So the removal of ammonia from body is ammonotalism. Removal of urea from body is ureotalism. Removal of uric acid from the body is known as uricotalism. That's what you need to keep in your mind, right, bache? That's what you need to keep in your mind. Okay, so now let's write the few points related to it. So, removal of ammonia is ammonotalism. Removal of urea is ureotalism. Removal of, yes, bache, removal of uric acid is uricotalism. So, let's talk about the types of animals. So, firstly, let's discuss about the ammonotalic organism. Ammonotalic organism, as the name is saying, that they are the one that will remove ammonia, ureotalism, ureotalic organism, and uricotalic organism, right? And other than that, bache, other wastes are also there. Like some organisms, they just excrete out some purines only, right? Some organisms, they excrete out amino acids. They are aminotalic. They excrete out amino acid. Some organisms, they show the dual excretion methods. Like let's say when they are in land, their waste will be different. When they are in water, their waste is different. So these are also the kind of discussions that we have. Okay, so firstly, let's start with the ammonotalic organism. Obviously, they will excrete out ammonia, and we will start with their requirement of water. So in the case of, yes, bache, in the case of a monotelic organism, the requirement of water is very high. See, that's how you have to remember. Overall, let me tell you that when we talk about the ammonia, ammonia will be excreted out in the form of ammonium ions. A for ammonia, A for aquatic. Aquatic organisms can afford to be a monotelic. Why? Because for the removal of ammonia, too much of water is required. Very large quantity of water is required. And ammonia will be eliminated from the body in the ammonium ions form by a diffusion only. Kidneys are not going to play any role there. So yes, to remove it from our body, a large amount of water is required, right? A large amount of water is required, and it's a very, very, very toxic, right? But it's a very toxic nitrogenous waste. One by one, we will discuss it. So requirement of water is very high here. It is, of course, it is lesser than ammonia. Right, here it is lesser than ammonia, and here it is least, right? So uricotelic organism, I'll give you the example of birds. You have seen birds excreta, birds potty, right? You have seen that, right? It's a very common affair. Like, let's say if you are sitting under a tree, sometimes birds, they do to check, right? Sometimes you are, all set, all ready to go out, or you are waiting for a cab under a tree. Again, the birds to check. They are waste. They don't, it's not like birds will secrete urine separately and the excreta separately. It's all same. It's all same. You can take the example of insects, right? So this waste is in the form of some pellets, right? Or in a kind of paste, Are you getting my point? So here the water requirement is least. Here the water requirement is least. Understood? Here the water requirement is least. So if I talk about the toxicity level, right? It is highly toxic. It is highly toxic. This one is highly toxic. It's very toxic. And of course, it is less toxic and it is least toxic. It is lesser than ammonia and this one is least toxic. Is that clear? Is that clear? This one is lesser than ammonia. This one is least toxic. Okay, bache. So now when you talk about the mechanism of excretion, right? So mechanism of excretion. So as I said, diffusion. Here it is the diffusion. Okay, bache. Okay. Right? So it will be done by diffusion across body surface, right? Or even like if you take the example of fishes, so gills in fish in the form of ammonium ion. So that is a PYQ. That is a PYQ, right? So your ammonia, it will be excreted out in the form of ammonium ions, right, bache? Right, bache? Yes or no? Yes or no? Yes or no? Now here, right, what's going to happen when the organism is ureotelic? It's simple. Bache, see, in our case also, if you take our example, in our case also this is going to happen, right? Just if in our liver, there is a liver cycle. Sorry, there is a urea cycle. In our liver, what is there? There is a urea cycle. That is also known as ornithine cycle, right? That is also known as ornithine cycle. And let me tell you, it is also known as Krebs Henle cycle as well. Yes. So citrulline arginine cycle as well. These are the another names here. Right? These are the another names here. Just say one Krebs cycle we also discussed there. Right? One Krebs cycle we discussed there. Na? We discussed in plant physiology. No, I'm not talking about that. Right? I'm not talking about that wala Krebs cycle. I'm talking about this. It's the urea cycle. Okay, it is the urea cycle. It is the urea cycle. It is taking place in liver. It is having the different names. It is known as Krebs Henle cycle. It is also known as your ornithine cycle or ornithine citrulline cycle, right? So ornithine, citrulline, they are also the amino acids, right? Non-protein forming amino acids there. Citrulline cycle it is. So it is the urea cycle, right? In simple words, it is the urea cycle which is taking place in our liver, Urea cycle it is. What's that? And it is Krebs Henle cycle, not the Krebs cycle that we study in the plant physiology. It is the Krebs Henle cycle. Is that clear? It is the Krebs Henle cycle. Is that clear? Yes. Is that clear? Sure. So urea, right, so basically here it is the mean nitrogen, like basically what is going to happen. So here you are talking about the involvement of liver. Chalo, I'll discuss it here. So basically what's going to happen? The ammonia, it will form in our body. That ammonia along with carbon dioxide in our liver cells, right? In our liver cells, that is, you are talking about the hepatocytes. What are you talking? You are talking about the hepatocytes. So in the hepatocytes, this ammonia and carbon dioxide, they are going to give us the urea. So that is how this urea will be eliminated from our body. Even if you are talking about the humans, yes, in our case also the story is same, because we are also ureotelic. So in our body, ammonia will form. That ammonia in the liver, it will combine with carbon dioxide. As I said, ammonia is highly toxic, right? So even in the case of plants, let me tell you, even in the case of plants, the ammonia will not accumulate. Plants will change it into another form because it is highly toxic. So ammonia and carbon dioxide together, right, in the liver cycle, I'm not talking about that complete cycle, right? As I said, it is the urea cycle, hence the Scrabs, Henle cycle, ornithine citrulline cycle, or simple ornithine cycle it is. So it will do what? It will form urea. It will do what? It will form urea. What will it do? It will form urea. Is that clear? So that urea will be eliminated from our body, right, bache? And kidneys are going to play the role there, right? Kidneys are going to play the role there. Is that clear? Is that clear? Right? Okay. So urea will be transported by blood plasma to kidney, and then it will be eliminated from our body. Okay, then it will be eliminated from our body. Okay, so carbon dioxide and ammonium ions in the presence of enzymes, they will give us these non-protein forming amino acids, and that is how it will be done. Understood? Understood? So liver will produce it, transported by plasma, removed by kidney. So liver will produce it, transported by plasma, removed by kidneys. Is that clear? Is that clear? Sure? Are you sure? Yes, please, are you sure? So here they are produced by purines in liver, or Malpighian tubule. Malpighian tubule is the excretory organ in the case of insects. In insects. So one insect type study is the part of our syllabus now. Cockroach, so you should know about it. So it is produced by purines in liver, or Malpighian tubules in, right, bache, or Malpighian tubules in insect. Is that clear? So how will it be removed from our body? How will it be removed from our body? In the form of paste or pellet, as I explained earlier. Right, in the form of paste or pellet, as I explained earlier. So ammonia, A for aquatic. Ammonia, A for aquatic, right? And yes, let me tell you why aquatic organisms or the lower organisms, in their case mainly they are ammonotelic. Bache, its formation requires less energy, right? That is also the point. That is also the point, that in the case of this ammonia, yes, I'm writing it here, that formation requires less energy. Formation requires less energy. Is that clear? Is that clear? Their formation, it requires less energy, and here more energy is required, right? And here maximum energy. So that's why it conserves more and more water. And these are the adaptations, right? These are the adaptations. Is that clear? These are the adaptations. Is that clear? So now let's focus on the examples, right? Verbally I'm telling you, A for ammonia, A for aquatic. So your aquatic insects, many bony fishes, right? Even your tadpole, aquatic amphibians. So cartilaginous fishes, bony fishes, ammonotelic. Cartilaginous fishes, ureotelic. Guys, exceptions overall are there. It is biology, the science of exceptions. Exceptions are there, but that's how you have to remember this. Okay. Okay. So ammonia, it's readily soluble. It is excreted by diffusion because it is soluble, of course. So diffusion across body surface or through gill surface in fish as ammonium ion. So this point is definitely very important, that as ammonium ion, it will be excreted out from our body. Okay. So as I said, examples are very important, right? Baki see, aquatic organisms, they are in water. Now they are taking a step towards the land habitat. So that is obvious now. For land habitat, they need that adaptation. Yes or no? Yes or no? They need that adaptation. They are moving to the land. They don't have that much excess of water. So if their excretory product, if it will be eliminated with a lot of water, it's not going to help. How will they fulfill the needs? Are you getting my point? How will they fulfill the needs? It will not be easy for them. Yes, please. It will not be easy for them. Yes, please. Henna, it is not easy for them. Definitely not. Definitely not. So that's the reason. That's the reason. So kidneys, they don't play any role in the removal of ammonium ions. So terrestrial adaptation needs the waste which can conserve more water. So mammals and many terrestrial amphibians, just here, when it comes to the example, let me use a different color here for the examples. So many bony fishes, aquatic amphibians and aquatic insects, right? Here, mammals and many terrestrial amphibians and marine fishes, they mainly excrete urea. So marine fishes is the term that they have used. Simply you can also write the word bony fishes here, right? Bony fishes, they are a monotelic. Bony fishes, they are a monotelic. Is that clear? Right? And cartilaginous fishes, they are mainly your bony fishes, they are amonotelic. Here you can write it, that is given here. So marine fishes, your cartilaginous fishes are mainly marine. But yes, it's not like that bony fishes cannot be. I'm just simplifying it because it is also in the animal kingdom, bony fishes, cartilaginous fishes. So on the basis of that, I'm telling you, bony fishes mainly or many, they are amonotelic. And cartilaginous fishes, many, they are ureotelic. Understood? Understood? So here in the NCERT also they have mentioned it that ammonia produced by metabolism will be converted into urea, right bache, and then it will be released into blood and is filtered or excreted out by the kidneys. Is that clear? So some amount of urea may be retained in the kidney. This line again. Some amount of urea may be retained in the kidney or some of the animals to maintain the osmolarity. Osmolarity is the solute concentration. As of now, you can just highlight it, but later on, I will explain this point. Okay? I will explain this point. So do not forget this point, that some amount of urea may be retained in the kidneys by some of the animals to maintain a desired osmolarity. Understood? Understood? So next example is for the uric acid. You know that high energy is required, least amount of water is required, it is least toxic. So your reptiles, birds, land snails and insects, they excrete the nitrogenous waste as uric acid, and that too in the form of pellet or the paste. Okay, so there is the minimum loss of water, so they are the uricotelic. Any doubt? Any doubt? Examples are there on your fingertips, yes or no? Examples are there on your fingertips, yes or no? We have to increase the speed as well, right? Okay, chaliye. So before starting the human kidney part, so let's talk about the different types of different organisms and their excretory organs. And obviously it depends upon nature of nitrogenous waste and the habitat. And of course, nature of nitrogenous waste is on the basis of habitat only. So, nature of nitrogenous waste formed, right? It is different and it depends upon what? Nature of nitrogenous waste form in different organism. Simple. The habitat will decide. Okay? Right? Habitat will decide the type of nitrogenous waste. Habitat will decide the type of nitrogenous waste. That's what you need to keep in your mind. Say for ammonia, those who are aquatic, they can afford, right, the excretion of ammonium ions. They have that. Okay, bache, they have that. So now on the basis of that, we have to discuss the different types of excretory organs. Okay, bache, we have to discuss different types of excretory organs. Again, I'll do the same thing. I'll write the phylum names here. Just say porifera. Right. I hope now you remember. Cnidaria, Ctenophora. Yes, please. Platyhelminths, Esculminths, Annelida. Arthropoda, Mollusca, Echinodermata, Hemichordata, and then there is Chordata. And then there is Chordata. Is that clear? So Porifera, Cnidaria, Ctenophora, Platyhelminth, Esculminth, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. These are the phylums. Right, bache? These are the phylums. So when you talk about it, right, just say from Porifera to, you can say that, yes, from Porifera, like in the case of Porifera, water canal system is there, right? So Porifera, Porifera, they have water canal system that will also help in excretion, right? In the case of Cnidaria also, you know that when we talk about these three phylum, we will not talk about any special respiratory organ, right? Sorry, excretory organ. We are not going to discuss about any special excretory organ. But when it is the platyhelminths, we have the flame cells, right? What do we have? We have the flame cells. So simply here we can say that excretory organs, they are absent. Excretory organs, they are absent, but nitrogenous waste, right, excretory organs are absent, but nitrogenous waste is eliminated through general body surface. It is eliminated through general body surface. Okay, now as I said, when it is the platyhelminths, what do you have? You have the flame cells. Why do we call them as flame cells? They look like it. The another word here is protonephridia. What's the another word that you can use here? Protonephridia and solenocytes. What do we call them? We call them as solenocytes. Is that clear? What do we call them? Solenocytes. In the case of Esculminth, H-shaped rennet cells are there. That also comes under the protonephridia wala category. So they are H-shaped rennet cells. What are they, bache? They are H-shaped rennet cells. Okay, H-shaped rennet cells are there for the excretion. Understood? Understood. Now it is Annelida. Earthworm is the example there. Wait, I'll write it there. Then it is Annelida. Earthworm is the example. Your Nereus is the example. Your leech is the example. So in the case of Annelida, you talk about the nephridia. Different type of nephridia. It's not protonephridia, it's the nephridia here. What is it? It is the nephridia here. Now in the case of arthropod, when you talk about the insects, in the case of insects, we talk about the Malpighian tubules. Right, bache? Malpighian tubules, that's what we have at the junction of midgut and hindgut. At the junction of midgut and hindgut, what do we have? Malpighian tubules are present, right? Malpighian tubules are present. Is that clear? Malpighian tubules are present, right, bache? Right, bache? And other than that, in the like class Insecta, same way class Arachnida is there. So when you are talking about the Arachnida, I'll give you one example, spider. Right, you can take the example of spider here. Which example you all can take? You can take the example of spider here. So in their case, coxal glands are present. Right, coxal glands are present. Then you can take the example of crustaceans. Right, I'll give you the example of prawn here. Prawn. So it is the green gland. Green gland, they'll help in excretion. So preen gland is different. Preen gland is different. It is green gland. What's that? It is green gland. Right? It is the green gland. So green gland in crustaceans, you can take the example of prawn. Okay. Then mollusca. When you talk about the mollusca, right, this is the second, arthropoda is the first, it is the largest phylum, mollusca is the second largest phylum. So they have organ of Bugenus. Right, they have organ of Bugenus, also known as Cabber's organ. That is for excretion. And in the case of mollusca, that's a PYQ. That's why I'm telling you, you know, Ctenidia, the gills, right? Ctenidium is singular, Ctenidia is plural. Ctenidium is singular, Ctenidia is plural. Yes, do you know that? Ctenidia is singular, Ctenidium is plural. So in mollusca, we have the Ctenidia as well. These are the feather-like gills, right? So they help in respiration as well as in excretion, right? What is unique about them? They will help in respiration as well as in excretion. As well as in excretion. What is it? It is mollusca. What is it? It is mollusca. So overall, you have the organ of Bujanus or Cabard's organ there, right, bache? Right? But that's what you can remember. That's what you can write down. Okay? That's what you can write down. Now, the next is mollusca. Then comes the echinodermata. Wow. So echinodermata, no heart, no heartless, brainless. Echinodermata. You know that heartless, brainless. That's how I remember. And even there is no excretory organ, right? You know, here water vascular system is there. Water vascular, water ambulacral system. Ambulacral system. So they do not have the excretory organ, right? The water, they do not have the excretory organ. So nitrogenous waste will diffuse out through the body surface. If there is no excretory organ, the nitrogenous waste will remove through the body surface. Then comes the hemichordata. In the case of hemichordata, we have the proboscis, right? Proboscis gland is there. Is that clear? Is that clear? So proboscis gland is there. What is there? Proboscis gland is there. Is that clear? Now, bache, when you talk about the vertebrata, you have the subphylums. Just if I talk about Urochordata, we have Urochordata, we have Cephalochordata, we have vertebrata. So phylum Chordata, I'm sorry. Phylum Chordata is having these three subphylums now. Phylum Chordata is having these three subphylums now. So when you people talk about Urochordata, so Urochordata mein you have the neural glands or nephrocytes. Neural glands or nephrocyte, nephron word, somewhere associated with excretion. That's how you can remember. Okay, that's how you can remember. This is what you have in Urochordata. Cephalochordata, and this is given in NCERT as well. So cephalochordates, they also have flame cells. What do they have? They have flame cells. It looks like flame. That's why we say they are the flame cells. Understood? And then other than that, bache, you will see the kidneys. But yeah, in the case of kidneys also, like there is evolution of kidney. Pronephric, mesonephric, metanephric kidney. Right? Pronephric, mesonephric and metanephric kidney. Okay, bache? Okay, bache? Ma'am is practicing 10 years. Mr. Ganesh, you can go for last 15 years ka question practice. That will be better. Okay, that will be better. So cephalochordates are there. So after that, what do we have? We have the kidney. And as I said, metanephric kidneys. This is the evolution. The advanced one is metanephric, like we have. Okay, so that's what you can write down. So again, let's revise it from the NCERT. So you can take the examples also. You can take the examples also. See, protonephridia, flame cells. As I said, we also call them as solenocytes when we discuss it in platyhelminths. So in platyhelminths, that is your flatworms. Right, that is your planaria. You know, planaria, also known as digasia. So rotifers, even cephalochordates like Amphioxus, they have protonephridia, right? Which is concerned with ionic and fluid volume regulation, that is osmoregulation. So nephridia you will see in the case of earthworms and other annelids, right? So it will also help in the removal of nitrogenous waste and it will maintain the salt and water balance. Then Malpighian tubules are the excretory organ of most of the insects, including cockroach. So cockroach is the part of our syllabus, so do not forget that. So where these Malpighian tubules are present? At the junction of the midgut and hindgut. At the junction of the midgut and hindgut. Okay. Then Malpighian tubules and the removal of nitrogenous waste. Then antennal glands or green glands. Yes, green gland is having another name. I forgot to tell you. It is in the crustaceans like prawn. Okay. Green glands is having another name. Sorry. Green gland is having another name. That is antennal glands. Okay, that is antennal glands. So that's what you need to know down. And now, because we have to discuss the kidney. So when you talk about the human excretory organ, what are we going to discuss here? When we talk about the human excretory organ, yes, we will talk about pair of kidneys, right? Then pair of ureter, yes or no? Pair of kidneys, I believe it is the simplest diagram. Pair of kidneys, pair of ureter, and then urinary bladder, which is single of course, and then urethra. That's it. That's what we have in the human excretory system. Okay, that is what we have in the human excretory system. Is that clear? Is that clear? Okay. Okay, Baki, one more thing, bache. So if you talk about the type of nitrogenous waste, that is also something that we can discuss. So you know that porifera, See, porifera to esculment, they all are amonotelic. Porifera to esculment, they all are amonotelic. When it comes to annelida, in the case of annelida, the one which are aquatic, they are amonotelic. The one which are terrestrial, they are also the ureotelic. They are also the ureotelic. I'm not saying uricotelic, they are also the ureotelic. Arthropoda is mainly your Arthropoda is again mainly your uric acid. Terrestrial forms are having uric acid, but aquatic forms can also have the ammonia. And this spider, it also excrete out your, yes, in the case of spider, there is a kind of it is not ammonia, it is not your uric acid. In the case of spider, it can be, the example is guanine. Guanine is what? It is the purine. Right, it is the purine, that's the example. Baki mollusca again, bache, the story is simple here. In the case of mollusca, if it is the aquatic form, then ammonia will be there. If it is terrestrial, then it will be uric acid. Right, then it will be uric acid. Of course, that is the thing that you should know. Baki bony fishes, right, mainly what do they have? Bony fishes, they will excrete out ammonia, but the one which are there in the marine water, they have different excretory waste. Okay, cartilaginous fishes, they are also having that urea. Okay, they are also having that urea. Okay. Okay, so that's how you can say. So sometimes land forms, they have urea, sometimes land forms, they have uric acid. Even in the case of birds, yes, even in the case of birds, let me write it here. Like insects, they also excrete out uric acid, right? Uric acid. And bache, in the case of amphibia, terrestrial forms will have urea, aquatic forms will excrete out ammonia. In the case of reptiles, I'm writing it randomly, not in a sequence. It will be uric acid. It will be uric acid. Is that clear? It will be uric acid, right, bache? And that too in the terrestrial form. Aquatic again, insect, right? Or mammals maybe, and that too in terrestrial forms, because aquatic will have insects. Ammonia. They will have ammonia. So that's the point. Done. That's the point. Baki human excretory system, that's what we need to discuss. And trust me, the diagram is very easy. That's how we draw it now. Okay, so pair of kidney, ureter, urinary bladder, and there is urethra. So what is it? What is it? It is actually the human excretory system. Human excretory system, and that's what we need to understand. That's what we need to know. So kidney, ureter, urinary bladder, and then there is urethra. So let me show you this. See, it's a dissected kidney, right, bache? Ye dekho, look at the diagram first, right? Look at the diagram first. Look at the diagram first. Human kidneys. See, this one, this kidney, bean-shaped structure, right? This tube-like ureter is there, right? This part here, this concavity here will be the hilum. This concavity here will be the hilum. Is that clear? This concavity here will be the hilum. Okay, bache? Okay, so renal artery veins are there. Look at the structure first, right? The cortex, the medulla. Look at the structures first, then I'll discuss it. It's good now? Yes, please, it's good. Hai na? Of course. It's very good. So let's discuss the structure here. So look at the diagrams, right? I believe if you understand this now, if you really observe it now, you'll get to know a lot. See this. So you have this kidney, you have this hilum, the ureter attached. So artery, if it is, so if it is artery, definitely it will carry the oxygenated blood. Veins, deoxygenated blood. These arteries further will divide into arterioles. Veins, they form the venules. Simple it is, right? Simple it is. So see the way it is present, right kidney, it is right kidney, this is also the right kidney, this is the left kidney. Okay, okay, so look at the diagrams first, then further I'll explain. It is amazing, indeed it is. Okay, it is. So examples, these diagrams, trust me, they are important, they are really helpful, they are important, they are really helpful. Okay, here देखो, few more. Right kidney, left kidney, look at their position as well. These are the blood vessels associated. This is your aorta, complete aorta. Okay, vena cava is also there. Vena cava is also there. These are the kidneys. Right kidney. IVC means inferior vena cava. So from a research paper, these images, they were available on Google, right? So they are authentic as well. So that's why I picked it up to show you that how things are there in our body, right? How do they look? So simple it is, you are talking about the kidneys, right? Kidney, I'll write the extra things also. So in kidneys, the excretory system is a pair of kidney, one pair of ureter, urinary bladder, urethra, done. So they are bache, reddish brown, bean-shaped structure, where it is situated with the level of last thoracic vertebrae and third lumbar vertebrae, right? And that too close to the inner body wall of the abdominal cavity. Don't worry, I'll use some extra terms here, right? Just like kidneys, they are retroperitoneal. They are retroperitoneal. I'll explain it as well, but firstly write down the information. Write down this information. So what are they saying? That it is situated in between the last thoracic vertebrae and third lumbar vertebrae. Just say the trick here is, now we have completed locomotion and movement, right? We have already completed that locomotion and movement part, right? Locomotion and movement part. So I believe you understand that we have CTLSC. That's the formula. CTLSC, 7, 12, 5, 5 and 4. These 5 are fused, these 4 are fused. So you have 7, 12, 5, 1 and 1. They formed after fusion. So last thoracic vertebrae means you are talking about T12. Then there will be L1, L2, L3, L4, L5. Right? If I am saying last thoracic vertebrae means I am talking about T12. Right? So they are saying it is present in between this last thoracic vertebrae and third lumbar vertebrae like this. Right? T12 to L3, it is present. That's what we are saying. And on the dorsal, right, dorsal abdominal wall, right, bache, close to the dorsal inner abdominal wall. Okay, dorsal inner abdominal wall. And what is written here? 10 to 12 centimeter in length, 5 to 7 centimeter in width, 2 to 3 centimeter in thickness, and on an average, this is the weight, 120 to 170 grams. 120 to 170 grams. That is written here, right? Approximately what's that? It is 120 to 170 grams. Doubt? Doubt? Doubt, 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 doubt. Okay. Okay. So that's what we need to continue. So you tell me any doubt here? You tell me any doubt here? Sure. So what are we saying? We are talking about the excretory organ. In our case, it is the pair of kidney, right? We are talking about the complete excretory system here, right? So right now we are talking about its appearance and position, and I told you about its position and everything, right? What I just said, I told you about its position and everything, and I hope this part is clear to you all, right? I hope this part is clear to you all, right? And where is it present? On the dorsal body wall, right? Near to the inner abdominal wall. And yes, one more thing I would like to mention here, just say in the starting itself when I was explaining you now osmoregulation, there I told you, right? Because exceptions we should always consider. Just I told you that osmoconformers, osmoregulators are there. The one that can regulate that thing, that can to regulate means, right, that we are having the control, we can regulate it. Right, we can maintain it. Okay, so most of the vertebrates, they are osmoregulator. Most of the vertebrates, they are osmoregulator. Just overall, if you talk about your plants, they are conformers. In organism and population, we used to read that. Now, so that portion is deleted. So plants, they are conformers. Overall, in animals, you can say approximately near about 1% of the animals, they are the, right, they are the regulators. Rest all are conformers. Right, the rest all are conformers. Okay, so when you are talking about the osmoregulators, most of the, like almost all vertebrates are the osmoregulators, but yes, there is one exception that I just forgot. except hagfish. Hagfish is the part of your syllabus, Mixin. It is given in your syllabus, Mixin, hagfish. Hagfish. It is not osmoregulator. It's a vertebrate, but it is not osmoregulator. Mixin, hagfish. And it doesn't even show the migration. It doesn't even show the migration. Is that clear? It doesn't even show the migration. Is that clear? Is that clear? Okay. I hope it is clear. Chaliye. So this is about the position. Understood? This is about the position, that this is how the kidneys are present. And it is a reddish brown bean-shaped organ. Okay, here you will see a kind of concavity. So as I said, we will discuss it. But first thing that you need to know here is, right, but how are they placed? They are placed towards the dorsal body wall. Right, bache? And it is just below the diaphragm. Right? How is it present? Dorsal side par, right, just below diaphragm. Diaphragm, you understand? Yes, diaphragm, do you understand that? It is also unique to mammals. Diaphragm, it is dividing your, it's a muscular tissue that is dividing your thoracic cavity and the abdominal cavity. Right, the thoracic cavity and the abdominal cavity. So just below the diaphragm, kidneys are present. Right, bache? Kidneys are present. Okay, so lower two diaphragm and lower two pairs of ribs. Lower two pairs of ribs protect them. So lower two pairs of ribs matlab, what are you talking? The last chapter that we have discussed is locomotion and movement. So I believe you understand this. Last two pair of ribs, if we are talking about, you're talking about the floating ribs, right, which are attached to the dorsally, which are attached to the vertebral column only. They are not even attached to the sternum. So floating ribs, they protect the kidney, right? Floating ribs, they protect the kidney. Is that clear? Floating ribs, they protect the kidney. Okay, bache? Okay, bache? Right? So when you talk about the kidney, just say, let's suppose, take the example, this is the kidney. I know it's not that easy for you to imagine it in this way, but still try to, if possible. Just say kidneys. In the starting itself, I have used the word that kidneys, they are what? They are retroperitoneal. That's the term that I have used. The kidneys, they are what? They are retroperitoneal. What is the meaning of that? They say peritoneum. Peritoneum, it is a serous membrane that covers the body organs. That covers the body organ. Now, when you talk about the kidneys from their front side, right? Kidneys from their front side means from their ventral side. Say if you talk about our body, here in our case, this side is our dorsal side, this side is our ventral side. This side is our dorsal side, this side is our ventral side. Are you getting my point? Sorry for this. Okay. So this side is our dorsal side, this side is our ventral side, this side is our lateral side. So dorsal, ventral, lateral. Dorsal, ventral, lateral. Okay. So kidneys, their front end, right? Just our front end is our ventral side now. So from their front side, right, they are covered with the peritoneum. Right? They are, from their front side, they are covered with peritoneum. Are you getting my point? From their front side, they are covered with peritoneum. Means if I have to tell you, just let's say that if this is the dorsal side of the kidney, this is the ventral. So here, right, you have the peritoneum. All the viscera is present here, right? Peritoneum is covering it, or peritoneum is also covering its ventral side. So, or we can see it in this way, that the kidney is just behind the peritoneum, or you can look at it in this way, that kidneys are just behind the peritoneum, right? Where these kidneys are just behind the peritoneum. Is that clear? Is that clear? So they are covered with what? They are covered with peritoneum. They are covered with what? They are covered with peritoneum on the front side only, right? Right. From their front side only, they are covered with peritoneum. That's why we are saying that they are retroperitoneal. Understood? Understood? Behind the peritoneum it is. Retroperitoneum means it is behind the, okay, it is behind the peritoneum. What's that? It is behind the peritoneum. Okay? Okay, bache? Sure? Sure. And they are, bache, slightly asymmetrical. Okay? Right, their position is also left kidney is slightly higher than, is placed slightly higher than the right one. So can you tell me why? Yes, can you tell me why? Why is it so? Why is it so? Why is it so? So kidneys, they are retroperitoneal in nature, right? So from their front side, from their ventral side, they are covered with the peritoneum. So they are slightly asymmetrical. That is, left kidney is placed slightly higher than the right one. Left kidney is placed slightly higher than the right one. Why is it so? Very good, very good. Right? Yes, please. You are right. Okay, you are right. So up to this part all clear? Up to this part all clear? Sure. Chaliye. So, our kidney is metanephric. So now when you look at the kidneys, right, on their medial side, this side, you will see a kind of concavity. What's the word? It is the concavity. It is the concavity. That's also PYQ. And this concavity is what we call it as hilum. We call it as hilum. So all the blood vessel, nerve vessel, lymph vessel, they will enter or leave from this part, right? It is the hilum, it is the concavity here. So all the blood vessels, they will enter or leave from this part. Is that clear? Is that clear? Right? So kidneys are metanephric. Kidneys are metanephric. Now let's talk about their inner structure. Okay, let's discuss about their inner structure. So when you talk about their inner structure, overall kidney, each kidney is made up of approximately, each kidney is made up of 1 million of nephrons. They are made up of what? They are made up of 1 million of nephrons. So now let's discuss their inner structure, bache. So they are having the microscopic uriniferous tubules or nephrons. Microscopic uriniferous tubules or nephrons there. They are microscopic. What's the word? They are microscopic. Okay, bache? They are microscopic. Or in each kidney, you can say approximately, let's pick up the NCERT line here only, having 1 million nephrons. Having 1 million nephrons. So we have 2 kidneys, so 2 million nephrons will be there, right? So they are what? They are microscopic. We cannot say the cell with the naked eyes. So it is the uriniferous tubule or the nephron, and we know that it is the functional, structural and the functional unit as well. What's that? It is the structural and the functional unit, right? It is the structural as well as the functional unit. That's what we know. Yes, please. That is what we know. Yes or no? So all these nephrons, they are held together by the connective tissue, right? All these nephrons so all nephrons are held together with connective tissue. Is that clear? CT here is connective tissue. So they are held together with connective tissue, means connective tissue is holding them all together, and these all are enveloped by a firm capsule, right? Okay, so connective tissue will hold all of them together, then a firm capsule will envelop them, right? And the firm capsule will envelop them, and that capsule is actually of what? It is white fibrous tissue. Again, structural organization, do revise it. So what's that? It is the white fibrous tissue. What's that? It is the white fibrous tissue. Understood? It is the white fibrous tissue. So all nephrons are held together with connective tissue, all enveloped by a firm capsule of white fibrous tissue, right? Of white fibrous tissue. Is that clear? Is that clear? Is that clear? Sure? Sure, sure, sure. Okay, so now imagine it like this. Just say connective tissue is holding it all together, right? And that is forming a kind of capsule. That capsule is of white fibrous tissue. Then, bache, there is the adipose tissue as well that is further covering it. Then there is what? Then there is the adipose tissue as well that is further covering it. Okay, adipose tissue as well, that is further covering it. And then there is one more, right, layer of connective tissue. We are calling it as the renal fascia. Just see this white colored one, you can say it is the white fibrous connective tissue that is making a capsule, and that capsule is holding all of it together. Okay, then what do we have here? This is the adipose tissue, adipose capsule or adipose tissue. Of course, adipose, fat. What is the function here? It is acting like the shock absorber. And outside this, there will be renal fascia. Fascia is the word, it is the connective tissue, and all these three are making the protective layers here. And this renal fascia, it helps the kidneys to get attached to the dorsal abdominal wall or inner abdominal wall. I'm writing it here, okay? So with the help of this renal fascia, it fixes the kidney. Inner abdominal wall. Done, students? Is it clear? Is it clear? That's how it is going to help. Understood? That's how it is going to help. Understood? Sure? Yes, please. Sure? So renal fascia will fixes the kidney to the inner abdominal wall. Okay? Okay? So overall in the kidney, when you look at the structure, when you look at its inner side, now you know about the layers. You know that when we look at the inner part here, what are we going to see? We'll see the inner cortex and the outer medulla. We will see the, outer cortex, inner medulla. Right, this outer part is what? It is the cortex part, and inner is the medulla here. Inner is the medulla here. M for middle part. Outer cortex, inner medulla. Outer cortex, inner medulla. We will discuss the nephron structure as well, but I believe you know this much. So let's say this is your nephron structure, right? Because they are microscopic and they are around 1 million in each kidney. So of course they are present. Where are they present? Where are they present? They are, yes, bache, they are present in the outer cortex. So when you look at this now, so even if you will see, just let's say if here there is a nephron here, so it's this loop-like structure will be there in the medulla. A part of collecting duct will be there in the medulla. We will discuss about it. So outer cortex, inner medulla, outer cortex, inner medulla, and here even in the medulla part here, bache, here even in the medulla part, what are we going to see? We'll see some pyramids-like structure. What are we going to see? We are going to see some pyramid-like structure. As the way I demarcated it, there is no such clear demarcation. There is no such clear demarcation when you look at the kidney structure. Right now, just to explain you this, I'm showing you like this. This is the outer cortex and this part is the medulla. But if you will look at the kidney structure, if you will look at its internal structure, then you will not say that there is a clear-cut demarcation. This is outer cortex and this is the inner medulla. Then you will not see this. Okay? Then you are not going to see this. Fine? It can be something like this. Okay, I told you already about the nephron position as well. Okay, something like this. Okay, something like this. So this is the outer cortex. This is the outer cortex and inner medulla. This is the outer cortex and inner medulla it is. Yes or no? Outer cortex and inner medulla it is. See, look at this. Look at this. So when you look at this medulla region, okay, when you look at this medulla region, right, here you will see these conical masses. Medulla mein you can see that there are, I'll write it here, 15 to 16 conical masses. What do we call them? They are known as medullary pyramids or renal pyramids. Medullary pyramids or the renal pyramids. What is it? What is it? It is the medullary pyramids or the renal pyramids. Is that clear? Is that clear? Yes, bache. Yes, bache. So when you look at these pyramids, na, just they are having this broad Like see, this broad base, it's their base, it is towards cortex side, and this narrow region, we use the word renal papilla for it. Do you know that? We use the word renal papilla for it. So these are the medullary pyramids. These are the medullary pyramids. So a narrow part which is towards the pelvis, it is the renal papilla. Broader part is towards the cortex pache. It is towards the cortex end, right? And narrow end is the renal papilla. Is that clear? Is that clear? Now this papilla will project into the cavity, right? This papilla will project into the cavity. It will project into the cavity of the minor calyx. Just say, let's suppose if this is the diagram, it is going to be something like this. It is going to be something like this. Like this papilla will project somewhere here, and it is what? It is the minor calyx. It is the minor calyx, right? Initially it is what? Minor calyx. Just say if I have to show you something like this, these minor calyx, they'll further form major calyx, right? Then it will open into the this part here. What is it? This funnel shaped structure here. This is the pelvis region. So calyx word is singular. Calyx word is singular. And when the word is calyces, it is plural. When the word is calyces, it is plural. When the word is calyces, it is plural. Okay? Okay? Done. So minor calyces, they join to form major calyces, right? And that major calyces, they will open into this funnel shaped area, right? They will open into this funnel shaped area, and this funnel shaped area is what? This is the renal pelvis. This is the renal pelvis. So 2 to 3, so minor calyces, they will join, they will form 2 to 3 major calyces, and that major calyces will further open into the renal pelvis, right? They will further open into the renal pelvis. Any doubt here? Yes, any doubt here? So this is a funnel shaped area, and then further the structure that we are making here is ureter. So again we will discuss it. So before that, let me tell you one more thing here. This is, see, a part of cortex. This is, let's suppose this is the part of cortex. Right, let's suppose this is the part of cortex. What's that? Right, this is the part of cortex. This is the part of cortex. So the part of cortex which is lying here in between these medullary pyramids, or in between this medulla, what do we call it? A part of cortex. It is not the part of medulla. It is the part of cortex which is extended here into the medulla. It is the part of cortex which is extended here into the medulla. Are you getting my point? It is what? It is the part of cortex which is extended here into the medulla. Right, part of cortex which is extended here into the medulla. What do we call it? We call it as medulla. We call it as column of Bertini. We call it as column of Bertini. Column of Bellini, duck of Bellini is different. It is column of Bertini. It is column of Bertini. Again, I am writing it here. Right, it is column of Bertini. It is the part of cortex which is extended into the medulla. It is the part of cortex which is extended into the medulla. That is a previous year question. Okay, that is a previous year question. Okay, bache? Okay, so between the pyramids, the cortex is extended into the medulla. You will call it as column of Bertini or renal column of Bertini. Any doubt? Any doubt? Are you sure? Are you sure? Pakka? So when we discuss the ureters, now sometimes we define it like this. Let's say this is the kidney. So ureter is entering into the kidney, and this is forming this funnel shaped structure that is known as renal pelvis. So sometimes we write it like this also. So do not get confused, right? Do not get confused here. Okay. So hilum I told you, the structure I told you, coverings I told you. So any doubt? Any doubt? Once again, this diagram. Do you have any doubt here? Yes, please. Do you have any doubt here? Look at this diagram. Look at this diagram. Yes, please, any doubt? See this. Now I think you can understand it in a better way, right? Some pyramid-like structures, some pyramid-like structures here, some pyramid-like structures here. Done? Okay, so ureter, from the hilum of each kidney, it emerged a cylinder whitish tube, that is ureter. है ना? From the hilum of each kidney, because we have the pair of ureter. That's why we are writing it like this. From the hilum of each kidney emerges a cylinder structure. What do we call it? Yes, bache. Stranded structure, yeah, stranded tube. What do we call it? We call it as ureter. We call it as ureter. Is that clear? Is that clear? Near about 28 centimeter long, bache, and they run downward along the muscles of the back, right, bache, and at the lower end of the abdomen, they bend inward into the urinary bladder. Right? We have seen this diagram, so I'm not going to write it. So we know that they will run downward, right, and somewhere there in the urinary bladder, they will just, right, in the urinary bladder, what's going to happen, bache? They will just bend inward into the urinary bladder. Now, what's the important point that we have to remember here? We need to know about the transitional epithelium. Transitional epithelium. This epithelium is also known as urothelium. In the chapter Structural Organization in Animals, we discuss about this type of epithelium. So transitional epithelium, which is also known as urothelium, my dear students, right? And it's a type of compound epithelium. What's that? It is a type of compound epithelium. That is a PYQ. So kindly listen to me carefully. What type of epithelium it is? It's a type of, yes, it's a type of compound epithelium. Compound epithelium means it is multi-layered, right? It is having many layers. Now, there is something special about this epithelium. It is the stretchable type of epithelium, right? We also call it as stretchable type of epithelium. Okay, so your ureter, your urinary bladder, it is lined with this. Your ureter, your urinary bladder, it is lined with this type of epithelium. What's going to happen? When you talk about this epithelium, as I said, it is multi-layered. Overall, these layers, they will lie on the basement membrane. Topmost layer is kind of globular. It is having the globular cells. It is having the globular cells. So I'll relate it with another example. Just say, when you talk about the, see, we get that urge to urinate, na? We get that urge to urinate, like we want to urinate, we want to pee, but we can control it for some time. Let's say you are in a class, right? You have the urge to urinate. Your body is giving you the signal you want to urinate, but you can hold it for some time, but later on you cannot. You can hold that urge, you can hold that thing for some time, but later on you cannot. Why? Why is it so? It is possible due to the transitional epithelium. It is possible due to the transitional epithelium. Just say, for an example, when you talk about the urinary bladder, your ureter, your urinary bladder, it is lined with urinary epithelium, which is a multi-layered structure where the topmost layer is having the globular cell. Now, when the urine will be filled, when urine will fill this, when urine is going to fill this urinary bladder. So when this urinary bladder is a little above the half filled, we start getting the urge to urinate. When this urinary bladder, it is little, like it is filled little above the, when it is little above the half, it is filled, we start getting that urge to urinate, right? But we can control it. Why? And say it's simple, because when the urine quantity will increase here, these globular cells, they will distend, they will become flat. These globular cells, they will distend, they will become flat. Right, so somewhere you can say that this capacity of urinary bladder will increase. It can hold more urine. It can hold more urine. Are you getting my point? Are you getting my point? Yes. So when we get the urge to discharge the urine, when it is little more than half of its volume. Right, bache? Right, bache? है ना? So and why this? So because this is the stretchable type of epithelium, so no doubt we will start getting that urge, but still it can hold it. Why? Because the globule, and let's say you are not able to go out to pee, to urine, for the urination. So what will happen? That globular cell will become distended. They will become flat, right? They will become flat. Okay. So that's the thing. So that's why we say the transitional epithelium is the stretchable one. That's why we say the transitional epithelium is the stretchable one. And overall, when you talk about the urinary bladder, you also talk about the detrusor, dartus muscle. Detrusor is there in the system. Dartus. Cremaster or dartus or detrusor? No, detrusor muscle that you discuss here. Yes. Yes, detrusor muscle that throw out something, that has a role in expelling out something. I'll mention it. जैसे ये देखो. See, that's how the ureter will bend inward towards the urinary bladder na. So when you talk about this urinary bladder, it is also a pear-shaped structure, right? It is also a pear-shaped structure. So when the term is pear-shaped, right, it is pyriform. When the term is like, what is the term we use for this pear-shaped structure? Pyriform. Okay, it is pyriform. So it is a pear-shaped structure. Where is it located? In the lower region of pelvic cavity. Okay, it is located in the lower region of pelvic cavity. Okay, it is located in the lower region of pelvic cavity. Okay, yeah, abdominal cavity. Okay, bache. So it is also thick, muscular, and having the distensible wall. So distensible wall, why? Because it is lined with transitional epithelium. It is lined with transitional epithelium. How is it lined? It is lined with transitional epithelium. Now you know the role of transitional epithelium. Is that clear? Is that clear? So musculature is well developed here, bache. Okay, so you have inner outer layers of fibers. Okay, so bladder is having the detrusor muscle, which is having the action of expelling a substance. So detrusor is the word. Detrusor muscle. So urinary bladder is having this muscle. So by mistake I also said nahi, dartus. So dartus muscle, this is something that we discuss in the male reproductive system. Cremaster muscle and male this dartus muscle. Right, so dartus we read, this word dartus and cremaster muscles we read in the male reproductive system, right? But this detrusor muscle, it is having the ability to expel out something. So it is there in the urinary bladder. It is there in the urinary bladder. Any doubt? It is there in the urinary bladder. Any doubt? Any doubt? Sure? Yes, bache? Okay, so now let's say if this is the bladder. So this lower part of the bladder, right? This lower part of the bladder, we call it as neck region, right? It is the neck of the bladder, like this lower part, right? This lower part. And it will further lead into urethra. It will further lead into urethra. So here we will see the sphincter muscles are present. Right, which muscles are present? Sphincter muscles are present. And when you talk about these sphincters, so sphincters, they are outer and inner. They are outer and inner. And these sphincters, they remain contracted. This point is important, huh? They remain contracted. So when they are contracted, they remain contracted to close. Right, to close that opening, right, so that urine will not pass out. So they will do what? So these sphincter, they are nothing, they are the muscle fibers. They remain contracted so that bladder will stay closed, right? To keep bladder closed, okay, to keep bladder closed. So they remain contracted to keep bladder closed. So sphincter, what is it? They are also the muscle fiber. Outer sphincter is there, inner sphincter is there. Inner, okay, inner, yes. Inner is involuntary. Remember this. And outer is voluntary. Means this one you can control with your own will, but when your urinary bladder is fully filled, right? We cannot control the passing out of urine, even if we don't want, because the inner sphincter will open, then this is involuntary. Understood? It is involuntary. So inner sphincter, it is involuntary. So if it is involuntary, can I say that if it is involuntary, right, you tell me, can I say that it is composed of smooth muscles? Because it is involuntary. Your smooth muscles are involuntary and outer, it is voluntary. So of course it is having the striated muscles, right? So inner one is also known as proximal and outer one is also known as distal. So these are the two sphincters which will be there in the neck of the bladder, which will lead into the urethra. Okay, bache, right? So spinal reflex is going to control it. Right, spinal reflex is going to control it. Remember that. Okay, remember that. Done. So external sphincter can suppress that urge. Right, so external sphincter can suppress that urge to pass out the urine. Right, bache. So cerebral cortex will direct the sphincters to relax. So when sphincters, bache, they will relax, so obviously there will be the contraction of the bladder muscles and urine will come out. Okay, so sphincters, when they are contracted, your urinary bladder is closed. So sphincters, when they will, right, when they will relax, the urinary bladder will open. Why? Because when these sphincters are relaxed, the cerebral cortex will tell the bladder muscles, then now you need to contract and you need to expel out the urine. Okay, now you need to contract and you need to expel out the urine. Okay, bache. Then comes the urethra. You know that this urinary bladder, from the lower part or the neck of the urinary bladder, it will lead to the exterior, right? So that is basically the urethra. Female mein, which is quite short, and males it is quite long, and females, they have the different urinary opening and the vaginal opening, right? But in the case of males, urinary opening, it is urinogenital opening, urinogenital aperture, urinogenital meatus it is. Okay, it is urinogenital aperture, urinogenital meatus, urinogenital opening. Is that clear? Is that clear? Chale. So now we have to discuss the inner part, like we have to discuss the structural and functional unit of kidney, that is the nephron. So the structure of nephron is important. Baki, let's come to the NCERT part. The things are quite simple here. That towards the center of the inner concave surface, it's a notch called hilum. Right, that inner concavity, it is hilum, through which the ureter, blood vessel, nerve vessels, they enter. Baki, inner to the hilum is a broad funnel shaped space, we call it as pelvis, which with the projections called calyces. So calyces, it's the plural term. So outer layer of the kidney is a tough capsule. Again I told you, see, connective, the nephrons, they'll held together with the help of connective tissue. Overall it will form a firm capsule that is made up of white fibrous tissue, and then there will be the adipose layer, then the renal fascia, that is the layer which will help that kidneys to get attached to the abdominal wall. Okay, so medulla is divided into conical masses, that is medullary pyramids, done and dusted. Column of Bertini, done and dusted. Right, so each kidney has nearly 1 million nephrons, right, and they are the functional units. They are the one that are going to filter out the blood and they are going to make the urine. Right, bache? Right, bache? So it is having two parts, the glomerulus and the renal tubule. So now let's discuss about the structure of nephron. Having what? Having two parts. Having what? Having two parts. So what are that two parts? Glomerulus and the renal tubule. Glomerulus and the renal tubule. So let's discuss the structure. Firstly, look at this diagram. Then I'll make it. We'll discuss it. And we will finish the chapter. So if nephrons diagram is clear, trust me, right? After that, urine formation we will discuss. So urine formation and countercurrent mechanism, they'll go hand in hand. You will understand it. Then the regulation of kidney function. These three are very, very, very important, right? These three things are literally very, very, very important. Okay, so just have a look of this diagram, then we will discuss it. So if there is any doubt, do let me know, guys. If there is any doubt, do let me know. Yes, please. Okay. 1 minute. Done, बच्चे? Sure. नहीं यार, break नहीं. चलो, let's discuss the nephron structure. No break. So see, this part, right, this tuft of capillaries, right, glomerulus, and then rest is the tubule. That's what we need to understand. That is what we need to understand. But before going forward, before even starting the nephron structure, I'll tell you the kind of blood circulation here, right, the blood flow here, right. We'll tell you what? We'll tell you about the blood flow. Cheek. We'll tell you about what? We'll tell you about the blood flow. Be ready for this, right? Be ready for it. So if there is any doubt up to this part, feel free to talk to me. Feel free to ask me the question. Yes, bache. Dekho. Overall, we know one thing. Heart. The trick is Lord. What does this trick means? Left side is having the oxygenated blood. Right side is having the deoxygenated blood. I believe you all understand this. Left side is having the oxygenated blood. Right side is having the deoxygenated blood. I believe you all understand this. What is the word here? It is the Lord. Okay. So left side of the heart, because it is having the oxygenated blood, you know that dorsal aorta will arise. We also call it as systemic aorta, right? And then further it will form different vessels which will innervate the body organs, because we have the closed type of circulatory system, right? What do we have? Because we have the closed type of circulatory system. So if I'm talking about the dorsal aorta, what do we have? If I'm talking about the dorsal aorta, what do we have? We have the oxygenated blood. Just say, let's suppose this is the dorsal aorta. So we are talking about the kidneys. So of course, we will discuss about the renal artery. Renal word is associated with kidney. So renal artery, it will give rise to, because we have the 1 million nephrons here, right? And in the paper, sometimes, yes, it happens when we read the questions, we mistake the nephron and the neuron part, right, bache? Right, bache? Is that clear? Yes, is that clear? So heart, dorsal aorta, then the renal artery, right? It is having the oxygenated blood. So these renal artery, right? Because it has to supply the blood to around 1 million nephrons. So then it will form arterioles. It will divide, it will form arterioles. That arterioles even, they'll form this tuft of capillary. We use the word glomerulus. Then efferent arteriole, that is also the terminology. So you can use it, right? Why am I telling you this? Because we have to discuss the nephron structure, and I don't want you people to get confused here in the terminologies here, that what is this efferent arteriole, what is this vasa recta? So you people should know that. Okay, so efferent arteriole is there. Then glomerulus. Then there will be glomerulus. Then there is efferent arterioles, A and E. This difference is important. Then there will be, you can say, the peritubular capillary network or the vasa recta. Will explain. As I said, I'll explain everything. Right now you make this flow chart. Peritubular capillary network or vasa rectitis. Then of course venules, that renal vein, and finally the inferior vena cava, and finally the heart will be there. Take. So peritubular capillaries or vasa recta, then there will be the vein, then there will be the, as I said, venules, renal vein that will pass its blood to inferior vena cava, and then the blood will pass to the heart. That's the complete blood supply. So heart will pump the oxygenated blood. Dorsal aorta is forming renal arteries, then efferent arterioles, glomerulus, efferent arteriole. There will be peritubular capillary network or vasa recta, and these are the capillaries. We'll show you the picture as well. Then venules, vein, and inferior vena cava, that will further pass it to the right auricle, right? That will further pass it to the right auricle, then right ventricle, and finally the lungs. We know it. So can I say that this side, right? That this side is having this oxygenated blood, and here you are talking about the deoxygenated blood. You know, oxygenated blood, deoxygenated blood, because your arteries, right? Your arteries, they carry the oxygenated blood. Veins, they carry the deoxygenated blood. Yes, there is an exception, that is your pulmonary artery and the pulmonary vein. This part clear? That is your pulmonary artery, that is the pulmonary vein. So ultimately in the urine formation, what's going to happen? There is the filtration of the blood, right? In the urine formation, what is going to happen? Ultimately, there is the filtration of the blood. We have to filter out the blood. So that is why these nephrons, they should have the blood supply, right? They should have the blood supply so that they can filter it. That's why that renal artery, for the dividing into arterioles. Now, when you know that setup, you know that this is the artery, they will form arterioles. Then capillaries are there, which are thin walled, right? Which are just having endothelium, so that exchange, so that diffusion and the exchange can be easy. Then venules and veins are there. That's why here we are writing it like this: peritubular capillary network of vasa recta, oxygenated, deoxygenated blood is there. That is why we are writing it like this. Any doubt? Any doubt? This is about the blood supply. Now, as I said, there are two parts in the nephron. So when you look at this, you draw this cup-shaped structure. It is my favorite diagram. Nephron, it is. So it is the functional unit of the kidneys. Structural functional unit of the kidney. It is. So in the paper, sometimes we just confuse the things. So it's nephron, not neuron. Okay, nephron. So these are the tubules here, and obviously you will see the blood like it will get the blood supply as well. 1 second. Okay, something like this. So focus on the diagram here, okay? In the case of nephron, it's very important to understand the diagram. Find something like this, something like this. Okay, so here in the structure of the nephron, this part here, right? This is the, like, how this nephron is getting the blood supply, which is this part. It is afferent arteriole, right? If you will look at it, its diameter, it's wider. It is wider, right? This tuft of capillaries here, it is glomerulus, right? This tuft of capillaries here, what is it? It is glomerulus. It is glomerulus. Now, right, from this cup-shaped structure, what is going out? What is going out from this cup-shaped structure? It is the efferent arteriole. The word is E for exit. The word is E for exit. It's important. You will see its diameter. It's narrower. This one is wider. This one is narrower. This information is important. Now, see this part here, this cup-shaped structure. What's that? It is the Bowman's capsule. Bowman's capsule. Okay. So glomerulus and this Bowman's capsule together, we call it as renal capsule or Malpighian capsule. These two things together, what do you call it? Renal capsule or Malpighian capsule. What do you call it? You call it as renal capsule or Malpighian capsule. Glomerulus and Bowman's capsule together. These two things. And this This part here you will see the first step of urine formation, the ultrafiltration or the glomerular filtration will occur. Okay? So this is the efferent arteriole that is narrow. It is important. Now this blood, what is it? This is a peritubular blood capillary network. This part, peritubular blood capillary network. And when you look at this tubule part, this is the lumen. This is the lumen of Bowman's capsule. This is the inner layer of Bowman's capsule, outer layer of Bowman's capsule. Now this part near to the Bowman's capsule, what is it? It is PCT. What is it? It is PCT. What is PCT? It is proximal convoluted. Convoluted means folded. So it is the proximal convoluted tubule. It is the proximal convoluted tubule. It is the proximal convoluted tubule. Understood? Proximal convoluted tubule. Now, this see, convolution means these foldings. Now this U-shaped structure, it is loop of Henle. It is loop of Henle. What's that? This U-shaped structure is loop of Henle. It is loop of Henle. Right? And here in this loop of Henle, this part, it is going down, it is descending. The blood will come like this, right? This is the descending limb. Thick descending limb, thin descending limb. This is the ascending limb. That's how the flow is going to be. That is how the flow is going to be. And this part here, it's DCT. What is DCT, students? Distal convoluted tubule. What is DCT? It's distal convoluted tubule, right? And then this part here, these are the collecting tubules, and this part here is the collecting duct. This part here is the collecting duct, right? Which is connected to many nephrons. This part here is the collecting duct. Okay, now when you look at this loop of Henle, right, it is the descending limb of loop of Henle, it is the ascending limb of loop of Henle, thick part, thin part. It is also covered with this, it is also covered with the blood capillary network. So what is it? Because it is U-shaped, we call it as vasa recta. We call it as vasa recta. What do we call it? We call it as vasa recta. This U-shaped capillary network. So it is the descending limb of loop of Henle, but in its vicinity, you will see ascending limb of vasa recta. This is the ascending limb of loop of Henle. In its vicinity, you will see this is the descending limb of vasa recta. That's what you need to remember. Again, just to clarify this portion, one second. It is important to understand, right? Or you can simply remember it. See this, right? That is what we have. So it is the loop of Henle, LOH, loop of Henle. Descending limb, ascending limb. The flow will be like this. Here the flow will be like this. Understand the diagram as I said. Now here it is surrounded by what? It is surrounded by vasa recta, this U-shape, the hairpin-like structure which is covering this loop of Henle. So this is descending limb of loop of Henle. It is the, right, the arrangement is like this only because it is the vasa recta. So of course students, here you will see the blood flow is there. It is the capillary network now. So blood flow is there. So descending limb of loop of Henle in its vicinity, right, you will see ascending limb, and here this is the descending limb. This is the descending limb. Is that clear? This is what? This is the descending limb. Fine. This is the descending limb. Fine. That's the structure here. That's the arrangement here. Okay. Okay. So now when you look at this part, this is the nephron. You'll see the arrangement is something like this. Like this part here is the cortex and this part will be the medulla of the kidney. medulla of the kidney. We'll show you other diagrams as well. So in the cortex, this woman's capsule, proximal convoluted tubule, distal convoluted tubule will be there. This loop of Henle, this U-shaped structure, it is there in the medulla. Some part of collecting duct is in the cortex, then rest is there in the medulla, right? That medullary pyramids, they have this. They have this. That's how the things are. Okay, that's how the things are. So understand this part. Here, dekho. This is also the diagram that we have. Fine, fine. So this woman's capsule and glomerulus together we call it as Malpighian capsule or renal capsule. Understood? So this is a PCT, proximal convoluted tubule, descending limb, ascending limb. Together they are forming Henle's loop, which is a U-shaped structure, right? And here you have distal convoluted tubule. And what's that? This is the vasa recta. So afferent arteriole, it is wider, its diameter is more, and efferent one is narrower. Understood? Diagram clear? Diagram clear? Here देखो, look at the arrangement. We will discuss the juxtamedullary, the cortical nephrons, but look at the arrangement here, right? The renal cortex, okay, and here you have the renal medulla. Fine, here you have the renal medulla. So this portion, see this, it is the renal cortex. Then here it is the medulla. Here it is the medulla. Look at the diagram. Look at the diagram. Now, bache, you will get a break for some time, right? For approximately 15 minutes. You have your meal and then we will meet. Okay, so let's meet at 8:45. 35. It's already 35, so 8:50. Fine. Let's have dinner break and then we will continue. Okay, then we will continue. Fine. Chalo. I hope I'm perfectly audible. Yes? Am I? I believe I'm perfectly audible. Okay, chaliye, let's continue, students. So this is all about the nephron structure. This is all about the blood flow there. Now let's discuss the parts, and now let's talk about the layers and about the urine formation. Okay, that's what we need to discuss. So up to this part, if there is any doubt, please feel free to ask the questions. Yes, please. Feel free to ask the questions. Done. So see, when you read this part from the NCERT, just 1 second. So see, that's how the points are given. Okay, जैसे ये देखो here. They have mentioned it that glomerulus, what's that? It's a tuft of capillaries. It's a kind of group of capillaries which is formed by the afferent arteriole, right? So what is afferent arteriole? It's a fine branched form of renal artery. Now you know the meaning here. Okay, so blood from the glomerulus, it will be carried away by an efferent arteriole. It will be carried away by an efferent arteriole. Okay, so the renal tubule, it begins with, right, with a double walled cup-like structure, that is woman's capsule, that is having the glomerulus, right, bache? So we call it as Malpighian capsule or the renal capsule or the Malpighian body. So tubule is having further, it is continues to form PCT, and it continues to form what? It continues to form the DCT. Okay. Okay, then Henle's loop and then the DCT. Fine, fine. So this is all about the nephron structure. Okay, bache. So see this, you can read this, that the Malpighian capsule, PCT and DCT of the nephron, they are situated in the cortical region of the kidney, whereas the loop of Henle, it dips into the medulla. It dips into the medulla. Okay, so when you are talking about the medulla, students, so in the medulla, no doubt you know that these are the conical pyramid, the conical masses, these pyramids are present. But of course, in between, you will also see the medullary fluid is there. In between, what will you see? You will see the medullary fluid is there. What will you see? You'll see the medullary fluid is there, or the interstitium, right? It is the medullary fluid or it is the interstitium. Okay, bache? Okay, done, done, done. Is it clear? Sure. Okay, so we have two types of now you know the structure. So on the basis of that, firstly let me explain this, and then we will talk about the cells and all that are lining this. So bache, we have two type of nephrons. We have cortical nephrons, right? We have cortical nephrons that are making majority of nephrons, and we have the juxtamedullary nephrons. Juxtamedullary apparatus is different. It is the juxtamedullary nephron. So what do we have, students? Yes, what do we have? We have the cortical nephrons and we have the juxtamedullary nephrons. What do we have, students? We have the cortical nephrons and we have the juxtamedullary nephrons. So when you talk about these cortical nephrons, as I said, they make majority of the nephrons, right? Right, so they constitute about 85% of the total, or you can say that somewhere around 75 to 85% of total nephrons. Right, 75 to 85% of total nephrons, but they are basically 10 to 15%, or you can simply say that they are the 15% of the total nephrons. Is that clear? 15% of the total nephrons. Okay, bache. So when you talk about the Malpighian capsule, right? When you talk about the Malpighian capsule, so Malpighian capsule means you are talking about the woman's capsule and the glomerulus here. It is close to the kidney surface. Right, I'll show you the diagram as well. It is close to the kidney surface. It is close to the kidney surface. Is that clear? But it is located at, but here it is located at the same thing. It is located at the junction of cortex and medulla. Okay, it is located at the junction of cortex as well as medulla. Okay, here you will see that there is a short loop of Henle, maybe or may not be covered with vasa recta. Okay, so loop of Henle, that is LOH, it's quite short and mostly confined to the cortex, right? It's short, mostly confined to cortex, means if it is quite short, it will be there in the cortex only, and right, and a very small part of it runs into medulla. It runs into medulla. It runs into medulla. Is that clear? Is that clear? But here the loop of Henle, it's quite long, and basically it is deeply embedded into a medulla. Here vasa recta, well developed, well developed. Here it is absent, either it is absent or reduced. Either it is absent or reduced. Is that clear? Absent or reduced. Is that clear? Is that clear? Here, when you talk about the peritubular capillary network, right? They are well developed. Right? What are they? They are well developed. But here they are not well developed. Fine. These are the two type of nephrons that we are going to study. So majority of nephron is what? It is the cortical nephron. So bache, see, when there is the water scarcity in our body, right bache? So at that time you will see that juxtamedullary nephrons, they are going to play the role. They are going to absorb. They are going to absorb the water as much as they can. Is that clear? Is that clear? Right? Okay. Okay. Take. So these are the two type of nephrons. So now look at the diagram here. Ye dekho, this is the cortical nephron. What kind of nephron it is? It is the cortical nephron, the short loop of Henle, and a part of it will dip into the medulla. But here, this loop of Henle is quite long. So how will it help? It help in absorption of the water as much as it can. So here you can see, when you look at this part, right, the Malpighian tubule part, you will see it. It is near to the kidney surface, near to that fibrous capsule part, right? And here you will see that it is at the junction of, okay, in the case of juxtamedullary nephron, you will see it is at the junction of cortex and medulla. Okay, so here this loop of Henle, it dips into the medulla, part of it will dips into the medulla, and here you can see it is deeply embedded there. And this part, what is it? It is the collecting duct. What is it? It is the collecting duct. So now when you look at it, right, just say, see, this is the U-shaped loop of Henle. So it is descending, right? What is happening? It is descending into the, right? What is happening here? See, this limb, it is descending into the medulla. And from here, it is ascending from medulla into the cortex. It is descending from cortex into the medulla. So the arrow shape is like this here. The flow of blood will be like this here. It is the filtrate. Right, not exactly the blood, it is the filtrate. So that will be the arrow shape. This part is clear or not? Yes, please. Is it clear or not? Is it clear or not? Take. So now we have to discuss the 3 steps, and along with it, we will also cover, right, we have to discuss the 3 steps here in the under the topic urine formation, right, but the question can come from this part. And here itself, we will also discuss that the kind of cells which are lining the nephrons. Okay, here itself, what will we are going to discuss? We will see the nephron, the kind of cells that are lining it. So urine formation, there are 3 steps, bache. There are 3 steps, and when you understand the nephron structure, parallelly you will get to know about it. The first is the glomerular filtration. In the book itself it is written, but I want you people to write it as well, which is also known as ultrafiltration, right? Which is also known as ultrafiltration. Then there is the tubular reabsorption and, sorry, there is the tubular reabsorption and the tubular secretion. What is there? There is a tubular reabsorption and there is the tubular secretion. That is what we have. Then there is the tubular secretion. Is that clear? Is that clear? These are the 3 steps. And of course, because nephron is the structural and the functional unit here, so of course the nephron is the one that is going to filter out the blood. And that's what we have to understand. Is that clear? That is what we have to understand. Is that clear? Is that clear? Sure. Okay, chale, let's start. let's start. Fine, let's start. So firstly, let's start with the women's capsule. Women's capsule. Let's start with the women's capsule. Okay. Yes. So the structure clear, right? The structure clear, right? Sure. So I will talk about the cells here. See, that is how the shape is, right? Firstly, understand it. The first step of, see, look at this. The structure is something like this. The structure here is something like this. Yes, everybody? The structure here is something like this. Yes. So here, what do you have? You have the outer layer of women's capsule, right? This one is the inner layer of women's capsule, and this one is the lumen of women's capsule, right? Lumen of women's capsule. And as I said, this part is the efferent arteriole here. This part is the efferent arteriole. Okay, so it is wider, it is narrower, and it is also important because it will create a pressure here, right? We will understand about it. We'll get to know about it. So now when you look at this outer layer, they are having very simple, right? Flat squamous cell, right? Simple layer. Simple means it will be uniserrated. It will be single layered. Okay. So when you talk about the outer wall here, it is just having the flat, right? It is just having the flattened squamous cells. Squamous means irregular shaped. Right, so flattened squamous cells are present. They are lining it. Now, when you talk about the inner layer, here you will see some specialized cell. Right, here you will see that there will be some specialized cell. There will be some special type of cells here. Okay, I'll explain it. Now, this is the glomerulus. This is the glomerulus. Means what? Glomerulus. Yes. What is it? It's a blood capillary. Okay, it is a blood capillary. So, if you remember this thing, you know that overall how the arrangement is. See, first of all, there will be a basement membrane. In the chapter structural organization in animal, we discuss it. There is a basement membrane. That basement membrane is epithelial tissue and the connective tissue. Both are going to release it. This basement membrane is not made up of any cell. It is non-living. It is acellular. And if you remember basement membrane, it is basal lamina, fibrous lamina. So I'm not discussing that, but I hope you remember that epithelial cells, they are going to lie on the woman's capsule. Okay, they are going to lie on the woman's capsule. So see, the epithelial cells, they will lie here on the woman's capsule. Sorry, epithelial cells, they will lie on basement membrane. By mistake, I was saying woman's capsule. So basically what I'm trying to say, that when you talk about the epithelial tissue, this epithelial tissue, it lies on the basement membrane. And under this basement membrane, here we have the connective tissue. That is how the arrangement is. So basement membrane, right? The part which is near to connective tissue, it is formed by connective tissue itself. The part which is near to epithelial tissue, it is formed by epithelial tissue itself. So this basement membrane is the acellular. It is having two parts, basal lamina and fibrous lamina. It is having that glycoproteins, polysaccharide, etc. Okay, now this is the epithelial tissue. I am talking about the capillary. In the capillary, you will see there will be only single layer of epithelial tissue. You also call it as endothelial, and there are pores in between. So this is about the glomerulus. This is about the blood capillary. The things are going to be like this here. But now when you are talking about the inner layer, of women's capsule, let's say this part. So here, as I said, the cells are present. We call them as podocytes. What do we call them, bache? We call them as podocytes. They are the kind of modified cells. What do we call them? We call them as podocytes because they will have some feet-like appendages. Are you getting my point? Why? Because they have some feet-like appendages. Poda word, right? It is associated with feet. So they have some feet-like appendages. Podocytes they are. So they are basically lining this inner layer of woman's capsule. Are you getting my point? They are basically lining this inner layer of woman's capsule. Are you getting my point? Right? Are you getting my point? These are that special cells. First of all, keep it in your mind. Right? So they have these kind of arrangements here. So first of all, tell me this part is clear to you or not. First of all, please tell me this part is clear to you or not, right? And of course, because it is also the kind of epithelial tissue, right? It is also the kind of epithelial tissue. Obviously, these cells are also lying on the basement membrane. Is that clear? Is that clear? Is that clear? Right? Sure. So when you talk about the inner layer of the woman's capsule, when you talk about the inner layer, see, if you will understand this part, trust me, everything will be easy for you, right? Do not forget that there are 3 steps in the urine formation: the glomerular filtration, the renal, the tubular, right? Glomerular filtration, the tubular reabsorption, and the tubular secretion is there. I know today I'm going slow, right? Yes, I am going slow. In the locomotion and movement, I was very, very, very fast. Today, I'm going a little bit slow. But that's okay. We have time. So please pay attention. So again, I'm saying the inner layer of women's capsule, what will you see here? You will see podocytes, right? You will see podocytes. So they are present in such a way, in such an intricate manner, that in between you will also see some slit pores, right? Right. In them, you will see some slit pores. Pores, you understand now? Openings are present, right, bache? So cells, they will lie on the basement membrane, and these podocytes, right, they are those who are forming the inner layer of the woman's capsule. They have some, right, they have some gaps, and we call them as slit pores. Now, when you are talking about the endothelial, right, that is the endothelial of the woman's endothelium of the glomerulus, right? Because glomerulus is a blood capillary. So that endothelial, they are also having some pores in between, right? They are also having some gaps in between, We call it as fenestrae. These are the 2 terms that you will understand. So these podocytes, they are also lying on the basement membrane. These endothelial cells, they are also lying on the basement membrane. So when you look at this particular part, of this nephron, right? Like this is the inner layer of woman's capsule. This is the glomerulus. First of all, they are in very close proximity, right? So these 2 layers, inner layer of woman's capsule, this endothelial here, right? Plus the basement membrane present in 2. Right, plus the basement membrane present in 2. This is the place from where the blood will filter out. This is just like a filtration membrane. So see, the blood will enter here from afferent arteriole, right? This is the glomerulus. So here the blood will get filtered. This is the ultrafiltration. The blood will get filtered here. It will enter here into the lumen, and the filtration membrane, it is having the endothelial of glomerulus. It is having the that podocyte of inner layer of woman's capsule, plus the basement membrane in 2, right? So these 3 are forming the filtration membrane through which the blood will filter out. I'll tell you the difference here also, why this difference is important. I'll tell you about the pressures, but tell me, this structure is clear to you or not? Just see, if you can focus, right? If you can focus, I'll use this diagram again. So when you look at this part, afferent arteriole wider, efferent arteriole comparatively narrow, and where, see, glomerular capillary, and this is the inner layer of woman's capsule. They are so close to each other, right? So look at the special cells here, podocytes here. Look at these special cells here. They are arranged in such a way that slit pores will be present here. Look at the glomerular capillaries. Look at the endothelial. They have this gaps here in between. We call them as endothelial. Right? So from here, the filtration of blood will occur. And outer layer of woman's capsule is having simple flat cells. So the blood will filter out here and it will pass to the lumen of the woman's capsule. Right? But blood will come, it will pass like this. Understood? Blood will come, it will pass like this. So see, podocytes, this glomerulus. This arrangement clear? The very first step is this arrangement. So one by one, I'll explain you the arrangement. One by one, I will also explain you, right, I will also explain you the nephron and the parts and the epithelium, everything. Because yes, question can come from this part. Okay, right? Okay, so this is the first step. Now see, this line is given ki on an average 1100 to 1200 ml of the blood is filtered out by the kidneys per minute, right? 1100 to 1200 ml of blood is filtered out by kidneys per minute, right, bache? And this is the 1/5 of the blood pumped out by each ventricle of heart in a minute. Do you understand this part? It's simple, bache. I'll explain it here only. See, heart. See, here in the case of heart, we'll talk about the cardiac output. How can we check that cardiac output? It is your stroke volume multiply heart rate. Stroke volume multiply by heart rate. So stroke volume is what? What is this stroke volume? It is the blood pumped by ventricle. It is the blood pumped by ventricle in per beat. okay, in per beat, okay, per beat, and that is 70 ml. And you know that on an average the healthy individual is having, right, on an average it is 72 times per minute are heartbeats, so it will be approximately 5040 ml, or can I say that approximately 5 liter, so right, approximately 5 liter, yeah, 5040 ml. So it's right, it's 1/5 will be, somewhere around 1100, 1200, yeah, 1000, 1100, right? So that is the meaning of this line here. So when they are saying that, when they are talking about the heart, right, that see, so heart per cardiac output means, right, that in a minute our heart beats for the 72 times. So it will pump this much of blood. So stroke volume is the blood pumped by a ventricle in per beat, by each ventricle in per beat. Heart rate, you know that 70 into 72, that's how we get it, right? So when you see the cardiac output, it is around 5000 ml or 5040 ml. So of course it's 1/5 is, right, this is the 1/5 of that. So it is the blood filtered by kidneys per minute. It is per minute, and it is the 1/5 of the blood pumped out by each ventricle of the heart in a minute. Now this part is clear or not? Done? Sure. So glomerular capillary blood pressure cause filtration of blood through 3 layers. I already told you about the 3 layers. Endothelium of the glomerular blood vessel, the epithelium of the Women's capsule, particularly you are talking about the inner layer of the Women's capsule, and the basement membrane between these, right? And the basement membrane present between these, epithelium of Women's capsule and the basement membrane between these 2 layers. Is that clear? So again you can read it. The epithelial cells of the Women's capsule, they are known as podocyte. This is a kind of modification, right, bache? Modification, like poda, feet like appendages somewhere they are, right? So they are arranged in such an intricate manner. They leave some minute spaces that are the slit pores or the filtration slits. Means that cells are arranged. Suppose, just to give you this example, imagine these are 2 podocytes. Just imagine these are 2 podocytes. So they are arranged in such an intricate manner that in between gaps are there now. So what are we calling it? We are calling it as slit pores. What are we calling it? We are calling it as slit pores, right, bache? Or we can also call it as filtration slits. So we can call it as slit pores, or we can also call it as filtration slit. So blood will be filtered from this side so finely through these membranes that almost all the constituents of the blood or the plasma except the proteins. So the word here is plasma. If I'll use the word blood here, so I will say some plasma protein and formed elements, right? I'll come to this. So this is the ultrafiltration. See, we are talking about the urine formation. We are discussing about the urine formation, and we are talking about the first step of urine formation, and that is ultrafiltration. So other than ultrafiltration, we can call it as glomerular filtration. Other than ultrafiltration, what can we call it? We can call it as glomerular filtration. What can we call it? We can call it as ultrafiltration. So what is happening here? See, just now you know the cells, now you know the arrangement. I believe you will be able to understand it in a better way now, because now you know what kind of cells are present there, how well they are arranged, the difference and the diameter, everything. So now blood will come here, blood will get filtered. It will pass here to the lumen. So when this blood will pass here to the lumen, what will we call it? We are going to call it as ultrafiltrate. What is the term that we are going to use here? We are going to use the ultrafiltrate, or glomerular filtrate is the term, right? Glomerular filtrate is the term. See, there are 3 steps in the urine formation, and let me explain you why that 3 steps are there. Dekho, this is the ultrafiltration. When this first step is going on, when this first step, that is ultrafiltration, when it occurs here, right, when this first step, that is ultrafiltration, when it is taking place, right, what is happening at this point? Like, it is not selective. This is not something selective. Are you getting my point? This is not something selective, that selectively the blood will be filtered out. No, No, like no, in a non-selective way, the blood will filter out. So here in this ultrafiltrate, right, its composition is same as plasma except some plasma proteins, because these plasma proteins, they cannot cross that filtration membrane. That's why they are not able to reach there. Now, if I will use the word blood here, you know that blood is having two components. Blood is having the plasma as well as the formed elements. So when we talk about the formed elements, what are we talking? Students, we are talking about the cells here, right? We are talking about the cells here. We are talking about the RBCs. We are talking about the WBCs. We are talking about the platelets here, right? What are we talking? We are talking about the RBCs. We are talking about the WBCs, and we are talking about the platelets here. We are talking about the RBCs. We are talking about the WBCs. We are talking about the platelets here. Is that clear? Is that clear? So blood is equals to plasma plus formed elements. So plasma plus formed elements. That is what we have. Plasma is that straw colored, yellow colored, that liquid which is having water, which is having other inorganic, organic things. You know that, right? So we are saying, because here in the filtrate, the blood cells should not be there. That will indicate an abnormality. That will indicate an abnormality. That is why we are using the word plasma here. This ultrafiltrate, its composition is same like plasma except some plasma proteins. Right? Except what? Except some plasma proteins. Right? Right? So basically when you talk about this ultrafiltrate, it is also having the glucose. Right? This ultrafiltrate, right? It is not the urine yet. It is not the urine yet, because two more steps are pending. So this ultrafiltrate, bache, it is even having the glucose. It is even having many amino acids. Right? Basically, it is having the glucose. It is having amino acid. It is having another extra things as well. Right? Right, it is also having some extra things, right? Extra required thing which we need in our body, So what is my point here? That when you talk about the ultrafiltrate, first of all, you will not call it urine, because it is not actually the urine. It is just a filtrate where we need to reabsorb many things, and we need to further add the ions just to balance the ionic, just to balance that pH, just to maintain the pH balance in our body, number one. Secondly, in this ultrafiltrate, the glucose is also there. And you know that in the urine, if there is the presence of glucose, that indicates the abnormality. That indicates glycosuria, right? That indicates glycosuria. And you know that it means that kidneys are not functioning well. It means that individual is suffering with diabetes mellitus. Right, so presence of urine in the and even the ketone bodies, right? It indicates that the individual is having diabetes mellitus. Is that clear? So that's what you need to keep in your mind. So why this filtration is taking place? The reason here is some pressures. This part is not given in NCERT, but it is important. Right, bache? So what is there? The reason here is pressure. So the effective filtration pressure that causes the ultrafiltration, it is determined by three pressures. There are three pressures which will decide it, right? So one is your glomerular filtration pressure, your glomerular hydrostatic pressure, colloidal osmotic pressure of blood, and capsular hydrostatic pressure. So what do you have? These are the three pressures, right? These are the three pressures that are responsible for that filtration. Number one is glomerular blood hydrostatic pressure. You can write it at GBHP, right? GBHP or simply GHP. So it means glomerular hydrostatic pressure or glomerular blood hydrostatic pressure, number one. Second is blood colloidal osmotic pressure. Then there is the capsular hydrostatic pressure. Now let me explain this to you. Dekho, it's simple. In the starting itself, I told you, right? In the starting, I told you that when you talk about the glomerulus, your efferent arteriole, it's wider than the efferent arteriole. That's why, bache, here the pressure will be created, and that the pressure will be created, and that pressure will help in the filtration. Am I right? And that pressure help in the filtration. right? How that pressure will help, students? That pressure will help in the filtration. Am I right? Yes, please. Yes, please. Yes, please. Take care. Okay, so what's going to happen? The blood will come here. It is wider, it is narrower, so the blood will get filtered here, right, bache, due to this pressure. So it is glomerular blood hydrostatic pressure, and it is a positive pressure. Positive pressure means it favors filtration, right? It favors filtration. I'll tell you all the definitions and everything, but firstly you tell me, is it clear? So because of this diameter difference, the pressure will be created here, and that pressure is going to, right, that pressure is going to do what? It is going to help in the filtration. So that is why we are saying that it favors filtration. That is why we are saying that it favors filtration. Is that clear? That is why we are saying that it favors filtration. Now, it's obvious, right? The blood is coming. The filtrate will pass here. But in the blood, that plasma, it is having the proteins, right? It's not like it contains some proteins, right? It contains some proteins. It is having some other ions. So in simple words, it is also having the solutes, right? It is also having the solute. Now you know the concept of osmosis, right? Let's suppose in the blood, the plasma, these proteins, right? These solutes, these things are present there. Even they don't want that further, that plasma to, the filtrate to go there in the lumen. They will resist the filtration. They will stop it, right? Next is what? Blood colloidal osmotic pressure. Like in the blood, we know that the solutes are present, right? So that will stop the filtration, right? It is simple, that will stop the filtration. Achha, one thing I would like to explain here, let's say this is a cell. If this cell is having more solute and it is having less solvent, so obviously it wants to absorb it wants to get like this is osmosis is going on in our body. If this cell is having more solute, less solvent, and obviously the surrounding cell is having less solute, more solvent, right? We know that osmosis will occur. That's how the things are. That's how the things are, right? That's how in the blood there is a definite composition. That's how the things are, because we have to maintain that balance. Why doctors, they advise us? Why doctors, they advise us that we should keep our body hydrated, right? Why the liquid is required in our body? Why is it so? Right? There are reasons. Yes, this is related to osmosis, right? Just say if you talk about the blood plasma, it is having the proteins. Albumins are present there. Right? That albumins, what will they do? They maintain the osmotic pressure of the blood. Because they are the proteins, the kind of solutes. They need the water. They have the water need, right? So they will keep on taking that water. Otherwise, that too much of water will be there in the tissue only. Tissue swelling can also occur, right? So these are the things, just to maintain that balance should be there. So of course, blood colloidal osmotic pressure, the proteins present here, they will be like, no, they will stop the filtration now. Here also in the lumen, you will have the filtrate. That filtrate will be like, I'm already here. Why are you giving me more? Right? It will also stop the filtration. So these are the certain pressures that will be created. Okay, so let me define it. It's very simple. You can see it here. So glomerular hydrostatic pressure, it is the blood pressure in the glomerular capillaries. And why is it so? Due to the difference, right? And why is it so, students? It is due to the difference in diameter of afferent and efferent arteriole. Understood? Due to the difference in diameter of afferent and efferent arteriole. Is that clear? It is due to the difference in diameter of afferent and efferent arteriole. Doubt? So bache, it is the main driving force that caused the filtration. So it is 60 to 75 mmHg. Then colloidal osmotic pressure, right? It is the osmotic pressure created in the blood of glomerular capillaries due to the plasma proteins. Okay, its value is 30 to 32. In some books, its value is even given in between 25 to 30 as well, right? In some books, its value is even given as 25 to 30. So this part clear? Yes, please. This part clear? Sure? Are you sure? This filtration pressure and all the things given here, they are important. So in some books, as I said, it is 30. It is given as 30. So it is due to the plasma proteins present. Then bache, capsular hydrostatic pressure, right? Then it is the capsular hydrostatic pressure. So it is, see, it is GHP. You can also write it as GBHP, glomerular hydrostatic pressure, yeah, glomerular blood hydrostatic pressure. Then it is blood colloidal osmotic pressure or colloidal osmotic pressure it is. So then comes the capsular hydrostatic pressure, right? Then comes the capsular hydrostatic pressure. So it is the pressure which is caused by the filtrate that reaches into the woman's capsule. That will also resist filtration, that will stop it. So basically this filtrate pressure is positive. Why? Because it is favoring filtration. Favoring filtration, these two are kind of negative because they are opposing filtration, So when we have to get the net or the effective filtration pressure, then glomerular blood hydrostatic pressure, from that you will subtract, right? You will minus blood colloidal osmotic pressure plus CHP, because these two things, they are not favoring, right? They are not favoring the filtration pressure, right? So this thing is favoring the filtration. So we will add it, then we have to subtract it from it. So ultimately this effective filtration pressure will be somewhere in between 10 to 25 mmHg. Is that clear? It is somewhere in between 10 to 25 mmHg. Is that clear? Is that clear? Please note down, right? It is somewhere around 10 to 25 mmHg. Okay? Okay? Sure? So this is the first step here. Okay, so positive pressure, negative pressure, effective filtration pressure, all clear. Effective filtration pressure, all clear. Done? Right, so this is the first step here. Okay, so the second step is, but before that I'll tell you about the filtration rate also. So, done. So that's the first step, and the rest, the things which are given here, like where it is, this one, right? This is, they are saying now that on an average 1100 to 1200 ml of blood is filtered by the kidneys per minute, which constitute roughly 1/5 of the blood pumped by each ventricle, right? By each ventricle. So it is actually the renal blood flow, right? This is actually what? It is the renal blood flow. It is actually the renal blood flow. Is that clear? It is what? It is the renal blood flow. Okay? Okay. Sure. Baki bache, with the, obviously from that you can also get the renal plasma flow, because you know that plasma is basically what? What is plasma? 45% or 55% of the blood? Plasma is what? Is it 45% or 55% of the blood? Tell me. Plasma is 55% of blood or 45%? Bache, again I'm repeating my question, right? Plasma is 55% or 45% of blood? Bache, it is 55%. So those who are writing 45%, guys, please don't waste your time. At least not for this particular year. Right? At least not for this particular year. That's something very basic. Right? That's something very basic. Chaliye, so this part clear? Right? This part clear? So the amount of filtrate which is formed by kidneys per minute, the amount of filtrate which is formed by kidneys per minute is called the GFR, glomerular filtration rate. What's that? It is the glomerular filtration rate, and in a healthy individual it is approximately 125 ml per minute, that is 180 liter per day. So the amount of filtrate that forms is 180 liter per day. But on the other hand, when you talk about the urine, like it varies from individual to individual, but it is near about 1.5 liter. So the filtrate formed is 180 liter, but the urine is 1.5 liter approximately. So of course 99% of the filtrate will be reabsorbed, because that filtrate formation is not selective. In the filtrate there are many things which body body needs to reabsorb. Like glucose is present, right? Other important amino acids are present, right, bache? So that's why, right? That's why. Okay. Okay. So this paragraph about the built-in mechanism and all, we will discuss later, right? So now let's move to the next part. So the lumen. In the lumen, what do we have? Yes. So in the lumen, what do we have? In the kidneys, in the lumen, what do we have? We have the filtrate, right? We have the filtrate, right? Right. So it will further pass to the PCT. Yes, it will further pass to the PCT. So try to understand the structure here. Let's say if this is the woman's capsule, there will be a small neck here. That neck will have ciliated epithelium, right? But now we will talk about the PCT, proximal convoluted tubule. I'll explain you the urine formation along with the cells here. So when you talk about the PCT, here you are going to see what kind of cell? You will see simple cuboidal, simple, it is again a PYQ, cuboidal, brush bordered epithelium. Simple cuboidal, brush bordered epithelium. What's that? Simple cuboidal, brush bordered epithelium. So simple means single layered, cuboidal means cube like cells. So when I'm saying brush bordered means the cells, they contain microvilli. When we are saying brush bordered, it means the cell, they are having what? The cells, they are having what? Right, bache? They are having microvilli. The cells, what do they have? They have microvilli. So microvilli, so that's why we are saying brush bordered epithelium. So microvilli, how will they help? Yes, microvilli, how will they help? They help in the absorption. They help in the absorption and they increases the surface area for absorption, right? They increases the surface area for absorption. So don't you think that if the PCT, if it is lined with the simple cuboidal brush bordered epithelium, then what is going to happen here? The reabsorption. So 70% to the 90%, right? They'll be reabsorbed here only. Here dekho. 70% to the 90%. Now come to the NCERT again. So comparison of the volume of, right? A comparison of the volume of filtrate formed per day, that is 180 liter per day, and the urine released is 1.5 liter, right? The urine release is 1.5 liter. So the filtrate formed is 180 liter per day and the urine formed is, urine released is 1.5 liter, right? So near about 99%, right? Near about 99% of the filtrate is reabsorbed here by the renal tubules, right? And this is the reabsorption. So obviously, right, because essential things are also there in the filtrate that body needs to reabsorb. Now the next paragraph, again important. Here we will stick to the NCERT only, right? You can even write down. So when it comes to the reabsorption, both type of reabsorption will occur. Like it will be done actively, it will be done passively. So when we are saying active reabsorption, you know that A for active, right? A for active, A for ATP, A for against. That's how you can remember. A for active, A for ATP, A for against. Right? That's how you can remember that A for ATP, A for active, A for against. Okay? A for ATP, A for active, A for against. That's what you can remember. Okay? So here when we are saying active absorption, so against the concentration gradient means low to high. Right? Low to high. Okay, low to high. So against the concentration gradient, the things will be reabsorbed. So of course here for this, energy is required, ATP is required, right? Right? And when you talk about the passive reabsorption, so you know that high to low, no ATP is required here. Okay, okay. Right? So glucose, please write down glucose, amino acid, sodium ions. They will be reabsorbed actively. Important MCQ. Glucose, right? Because if glucose is present in the urine, that will result in glycosuria. It means the diabetes mellitus, right? It means the kidneys are not functioning well. So glucose, even the amino acids, even the sodium ions, they will be absorbed actively. Nitrogenous waste, you're talking about the urea here. And remember, in the starting we have read one line that some amount of the urea will still be there in the medullary fluid to maintain the osmolarity. Slowly you will understand that part too. Okay. So urea are absorbed by passive transport. Chloride ions are absorbed by passive transport. Water is reabsorbed by passive transport. Are you getting my point? Right? Right? So in the initial segment of the nephron, this is going to happen. First of all, tell me this part clear. First of all, tell me this part clear. So it is regarding the reabsorption. I'll tell you about the secretion as well. But because we are moving one by one. So neck of the woman's capsule, it is having ciliated cuboidal epithelium, but PCT. What kind of epithelium is there in the PCT? What kind of epithelium is there in the PCT? Right, bache? It is brush bordered, simple cuboidal brush bordered epithelium. Right? Right? So it is simple cuboidal brush bordered epithelium. It is simple cuboidal brush bordered epithelium. Is that clear? So this is the PCT. Rest you know that. You know that this is your loop of Henle, the thick limb, the thin limb there. Right? In the NCERT, in that detail it is not given. But trust me, when you read good books, right, nice books by foreign authors, right, and books of your higher studies, you will find that the kind of epithelium is lining the thick limb of the descending limb, the thin limb of the descending limb, plus this thick limb is permeable to this and that, the thin limb is permeable to this and that. But here, because we are following the NCERT, so we will go accordingly. So you know that it is the descending limb, you know that it is the ascending limb. Oh, sorry, my bad. Descending, D for down, ascending, up. Right? So again I'm repeating this. Descending limb of loop of Henle, in its vicinity you will see there is the ascending limb of vasa recta. This is the ascending limb of loop of Henle. Here you will see descending limb of vasa recta. Why am I telling you this? Because this is something that we need to know. This is something we need to know in countercurrent mechanism. Now when you talk about this part, right, the descending limb. Yes, when you talk about this part, descending limb, it is having thick and thin part. It is having thick and thin part. Right, thick and thin part. So overall this thick part, bache, it is having same, right, it is having simple cuboidal epithelium. It is not brush bordered. This is extra information. It is having simple cuboidal epithelium. Right, this is extra information. You want to write it, you can. You want to drop it, you can. This part is the thin, and here you will have simple squamous. Squamous means flat and irregular cell. Right, so here you will have simple squamous epithelium. So here you will see flat and irregular cells. Here you will see single layered cube-like cells. Okay, single layered cube-like cells. Okay. Okay. Same is the case here. Here also you will see simple cuboidal. Here you will see simple flat cells. Now, what we need to remember when you talk about the descending limb, right? When you talk about the descending limb of loop of Henle, bache, it is permeable to water, right? It is permeable to water, but impermeable to electrolytes, or you can say that to solutes. Okay, or you can say that to solutes. So what's the word here? What's the word here? So descending limb of loop of Henle, it is permeable. This information is very important. This topic is very important. So it is permeable to water, but it is, yes, it is permeable to water but impermeable to electrolytes. Write it down. Write it down. Yes, it is permeable to water, but it is impermeable to electrolytes. It is permeable to water, but it is impermeable to electrolytes. Take. So see, when the filtrate, when it will pass, when the filtrate, when it will pass from this PCT then through the descending limb. But if water will come out in the surrounding, no doubt you have these capillaries plus interstitial fluid is there, medullary fluid is there, right? Because this part is in the medulla, you know it very well. You know the position. This part will be there in the medulla. So obviously if water is coming out, that will be added there into the medullary fluid. As of now, so this is what you can understand. Rest I will explain it in the countercurrent mechanism. So basically it will pass into the surrounding. It will pass into the surrounding. So imagine when the filtrate will come down, right? When the filtrate, it is coming down and the water, right, and this limb, it is permeable. Just in some books it is written ki thick limb. We are not talking about the thick limb, we are just discussing the thin limb, that it is a thin limb from where the water will come out. But theek hai, anyhow, let's focus on the NCERT. And it's simple that when you are talking about the descending limb, right, that when you are talking about the descending limb, the water will come out, right? The water will come out. Are you getting my point? The water will come out. Okay, so because it is permeable to water. Permeable to water means it can allow the movement of water only, but not the electrolytes, not the solute. So when water will come out, don't you think this filtrate will become concentrated by the time it reaches here? By the time it reaches here at the base, don't you think the filtrate, it will be concentrated? Yes, please. Yes, please. Don't you think that this filtrate, it will become concentrated? Because see what is happening, water is coming out. So by the time filtrate will reach here, it will be concentrated. Just imagine there is a solution. In that solution, solute and solvent is there. You are taking out the solvent, right? The remaining portion will be solute and some amount of solvent. So matlab that solution will be concentrated now, right? 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 Okay, now, okay, fine. Now your solute will become, by the time it reaches here, it will become concentrated. Now when you look at this particular part, right, the ascending limb, see from this side, from the thin side, passively, you will see passively, like, matlab, this AL of loop of Henle, ascending limb of loop of Henle, it is permeable to solute, electrolytes, whatever you want to write, but impermeable to water, strictly impermeable to water. Water movement will not occur across it. Fine, water movement will not occur across it. Is that clear? Is that clear? Yes. Is that clear? Right, so it is passively. It is passively. Passively what will happen? That electrolytes will come out, but water is not. Right, water is not. And here you will see some actively, you know, that NaCl and all, they'll come out. Okay, actively they will come out. So ultimately from this side, what will happen? The solute will be out. So now imagine, there is a solution. This is the concentrated one. It is having the solute and it is having very less solvent. Now, if you will take out the solute from it, right, if you will do what? If you will take out the solute from it, again it will become diluted, don't you think so? Because you are decreasing the solute amount there. You are decreasing the solute amount there. Yes or no? Yes or no? You tell me. Yes or no? Yes. So exactly. So again it will become dilute. Now when you talk about the DCT, what is this DCT? Distal convoluted tubule. Right? When you talk about the DCT, what is this DCT? It is distal convoluted tubule and it is also having the cuboidal epithelial cells. It is also having the cuboidal epithelial cells. Right? What kind of cells are there? Cuboidal, simple cuboidal epithelium. Now again the word is not brush bordered bache, but here there is conditional reabsorption. What is the word that we are going to use here? There is the conditional reabsorption of Na positive and water, right, etc. So conditional reabsorption means when required. So that part you will better understand when you will discuss the regulation of kidney function, that's how the aldosterone helps and how all that things are helped. So there is the conditional reabsorption, matlab when required it will be done. Then comes the collecting duct and the collecting tubule, collecting tubule and the collecting duct. Collecting duct se bhi bache, water can be reabsorbed. So then you will use the word conditional reabsorption. Now let me explain you this. ठीक, let me explain you this, and then finally we will talk about the tubular secretion. ठीक है, I have merged these two topics all together, but trust me, it will be easy. देखो, reabsorption is minimum in its ascending limb. Reabsorption is minimum in the ascending limb. Maximum is there in PCT. Minimum is there is ascending limb. But this region means loop of Henle or this ascending limb. It helps in high osmolarity of the interstitial fluid. Right? It is helping in high osmolarity of the interstitial fluid. Osmolarity, solute concentration you are talking about. So you are saying that overall when you talk about the ascending limb, again, important MCQ it is. So again, when you are talking about the ascending limb, right? Again, when you are talking about the ascending limb, right, bache? So in the case of ascending limb, it's very simple, bache. It is showing the minimum reabsorption. The reabsorption is minimum there in comparison to the other parts, right? But it is having a very important role in maintaining the solute concentration. It is having a very important role in maintaining the solute concentration. Okay, in maintaining the solute concentration. Are you getting my point? Are you getting my point? Because you know na, from here, actively or passively, the solute will be reabsorbed. The solute will be reabsorbed. So you better use the word solute, leave the word electrolyte. Right here, somewhere I have mentioned, na, you use the word solute only. You stick to that word only. Okay. Done? Right? Okay, so the descending limb of loop of Henle, it is permeable to water but almost impermeable to electrolytes. It is permeable to water but it is almost impermeable to electrolyte. Mr. Jyotish, you can type as much as you can. I'm not going to tell you that Dream Crash Course Batch Test syllabus. Attend the classes, you'll get to know about the syllabus. Simple. Done, students? So it is almost impermeable to electrolytes. So when the, see, this concentrate the filtrate as it moves down. Okay, so descending loop of Henle, I told you already, permeable to water. So when the filtrate will go down, right, it will concentrate the filtrate. And ascending limb, because it is impermeable to water, but allows the transport of electrolytes actively or passively. So it will concentrate the concentrated filtrate will again become diluted due to this, right? Right? So read about it. Yes, please. Read about it. Read it again. If there is doubt, do let me know. Read it again. If there is doubt, do let me know. Yes, please. But say some fire emojis in the chat section if it is clear to you. Tell me quickly. Yes. Yes, everybody, then I'll start. So in the DCT, what's going to happen? As I said, there is the conditional reabsorption of sodium and water, right? So it is capable of reabsorption of bicarbonate as well. That's why I mentioned the word etc. there, right? And selective secretion of hydrogen and potassium. So reabsorption, you are clear what will be reabsorbed. Now the third step of urine formation is tubular secretion. Matlab, it's like you can imagine the things like that today you have decided you are going to. Matlab, this is the example that I use again and again. That's it. Today you have decided that you you are going to clean your room. Okay, let's say today you have decided that you are going to clean your room. Okay, so because you are going to clean your room, this is what you have decided. So firstly we are like, chalo theek hai, I have to clear everything, so I'll put everything outside. Then you'll do all that brooming and everything, and then you will be like, theek hai, fine, I need this stuff, I need that stuff. You'll bring it back, you'll keep it, and then again you will check, okay, fine, these are the things, you know, I can keep it. Again you will realize ki these are the things that I can throw, right? I can throw, I don't need it. It's something like that, right, something like that. Are you getting my point? So same as the thing here. So firstly, normally, randomly, like non-selectively, that is the appropriate word, non-selectively the filtration will occur, then the reabsorption will be there from that filtrate, and then again, now because reabsorption is there, we have to, the kidneys, they need to maintain the pH balance, the ionic balance in the body. So what's going to happen next? So tubular secretion will be there. So when there is the tubular secretion, H positive, K positive, right, they will be added back into that filtrate. Okay, bache? Right, even the ammonia. So now what you need to remember, because they are like regarding new NCERT, old NCERT, some confusion was there na. So because we have to stick to the NCERT, then mark it accordingly. So when you talk about the tubular cells, they secrete H positive, K positive and ammonia into the filtrate. It is important step in urine formation because it maintains ionic and acid balance. So H positive, K positive, ammonia. Take, and here also when you are talking, that was for PCT. Now you are talking about the DCT. Here they have mentioned H positive, K positive, ammonia, right? H positive, K positive, ammonia. Look at this diagram. Here देखो, H positive is added, ammonia is added, and here K positive is added, H positive is added, right? So that's the difference. Okay. So again, NCERT, what NCERT is saying, that during urine formation, tubular cells, they add H positive, K positive and ammonia. They add H positive, K positive and ammonia. What do they add? They add H positive, K positive and ammonia. What do they add? Yes, please. They add H positive, K positive and ammonia. H positive, K positive and ammonia into the filtrate. Take, and here also, Henle's loop and then DCT. They have mentioned H positive, K positive and ammonia, but look at the diagram here, the way it is mentioned, right? See, bicarbonate, reabsorption, NaCl, reabsorption, water, nutrients, but K, here they are showing K positive ka reabsorption. Here they are showing, this arrow is showing secretion here. So that's how it is. That's what you need to focus, that's what you need to remember. Understood? Yes, please. Done. Okay, sure? Are you sure? Is it clear or not? Is it clear or not? Pakka? So focus here. Baki H2 reabsorbed, NaCl reabsorbed, that's how it is, right? That's how it is. And let me explain you the mechanism of concentration of urine, right? So I'll explain this thing also. Just allow me some time. I will explain this thing also. Allow me some time. Take. Then there is the collecting duct, bache. So you know that this is long duct which extends from cortex of the kidney to the inner parts of the medulla, right? So large amount of the water could be reabsorbed from this region, right? Could be reabsorbed from this region, means if required, if there is a water scarcity in the body and if we need to make more and more concentrated urine, then yes, bache, right? Then what is the point here? Right, bache? First of all, 1 minute ruko. See, Jyotish, you want me to continue this class or not? I'll end it otherwise, right? You are not attending the regular classes. In the regular classes, I have already discussed the test syllabus, right? You don't know it. And here in the YouTube, you are wasting your time, right? From last 20 to 25 minutes, you are sending the same message. Means you are that free. Do you really think you will be able to qualify the NEET examination with such kind of attitude? Huh? I have mentioned you the test date. I told you that check the batch, you'll get to know about it. You don't want to check it and you are just wasting the time here. What is this? If physics syllabus has changed, this is not physics class. Then discuss these things in the class itself. Now, why are you spamming here? I'll ask my team to block you. So this long dark extends from the cortex of the kidney to the inner parts of the medulla. You know the position, right? So large amount of water could be reabsorbed from this region to produce a concentrated urine. Okay, so some amount of urea, it also allows to pass into the medullary fluid to keep up the osmolarity. That's a fact. That's a fact. That's a very famous question, that your NaCl and urea, right? Your NaCl and urea, what they do? They maintain the osmolarity. They maintain the osmolarity of medullary fluid. These two are responsible for that. Let me explain you, right? Let me explain you the countercurrent mechanism. You'll understand it, right? Right? Okay? Fine. You will understand it because this is the next topic that we are going to understand. Okay? Then there is the regulation of kidney function and micturition. So main topic is these two, na, mechanism of the countercurrent mechanism and regulation of kidney function. These are the two main topics. Baki to bas disorders and other organs which help there. Take. We'll discuss it. So now let's talk about the mechanism of concentration of filtrate. But before going forward, just close your eyes for a minute and try to recall the things. Okay? Okay? Just close your eyes for a minute and try to recall it. Quick. Try to recall it. Ki haan ji, theek hai, it's clear to me, right? It's clear to me that fine, from the descending limb, the water will be reabsorbed. From the ascending limb, the solute will be reabsorbed. If it is clear to you, do let me know in the chat section. Then I'll continue this part. Okay, then I will continue this part. All of you, please. Done? Chaliye, so what do you think that what is the need to concentrate the filtrate, right? What is the need to concentrate the filtrate? What is the need? Why it is required? Why? Why it is required? Yes, please. To lose less water. Seriously, anything else? Yes, anything else? Yes, students, anything else? Okay. See, we are going to talk about the countercurrent mechanism, very important topic, and I have seen some students considering it as a very difficult topic, but it is actually not. It is a very easy topic. Okay? So that's what I'm going to explain. Fine, that's what I'm going to explain. So I believe the rest everything is clear to you. Okay, the urine part, the everything is clear now. So before going forward, I'll tell you the urine composition. When you talk about the urine, you know that urine output is 1.5 liter per day, right? The filtrate formed is 180 liter per day. So 99% of the filtrate is reabsorbed. Okay, right? 99% of the filtrate is what? 99% of the filtrate is reabsorbed. Now, when you are talking about the urine, bache, so urine mein, like, no doubt water is there, right? No doubt water is there. So on an average, right, you know that it is 1 to 1.5 liter per day, 1 to 1.5 liter per day, but it depends upon our diet. It depends the amount of water intake and everything. This is on an average. This is on an average. And on an average, remember this thing, on an average, 25 to 30 gram of urea is excreted. You know that 25 to 30 gram of urea is excreted out per day. Right? It is excreted out per day. Are you getting my point? 25 to 30 gram of urea is excreted out per day. Is that clear? Is that clear? Urine, when you talk about the pH there, right, normally it is somewhere around pH is 6, but again, it depends upon your diet, right? It depends upon your intake. pH is near about 6, but it depends upon your diet, it depends upon your intake. Okay? So before starting the countercurrent mechanism, again, I'll repeat the same thing. Again, I will repeat the same thing, and I really want you guys to keep it in your mind. At least till NEET examination. Okay, at least till NEET examination, I want you people to keep it in your mind. Right? Take. That's the thing. That's the diagram. Do not forget this diagram, and as I said, I have repeated it multiple times. This is your loop of Henle. Descending limb of loop of Henle, so the flow will be like this. Ascending limb of loop of Henle, so the flow will be like this. This is the ascending limb of your vasa recta. The flow will be like this, and this is the descending limb of your vasa recta. The flow will be like this. Now we are going to talk about the countercurrent mechanism, and that countercurrent mechanism is a unique adaptation that we have, right? It is a kind of terrestrial adaptation also. Birds are mammals and having this, right, bache? Birds and, right, bache? Birds are and mammals, they are having this. So, right, what is the thing that mammals, they have the ability to produce a concentrated urine. When the urine is concentrated means it will have less water, and when it is possible, just for an example, if your water intake is less, right, you know that the urine, when you urinate, that urine is like yellowish, right, dark yellow, very dark. Right, we get to know that, okay, our urine is concentrated. And when you drink a lot of water and when you urinate, because then you urinate very frequently, okay, when you drink a lot of water, you urinate very frequently, and then you see that the color is not that dark yellow, and plus sometimes it is just like water only, Why? Because your body is having sufficient amount of blood, the water, right? Your body volumes are good. Right, body fluid volumes are good. You have the sufficient water in your body. But when there is a scarcity of water, then body will try to conserve more and more water, and due to which it will make the concentrated urine. Right, so that we will explain under the countercurrent mechanism. Okay, so Henle's loop and vasa recta. Henle's loop, your loop of Henle, Henle's loop, one and the same thing, and vasa recta is going to play a very important role there, right? So can I say that because in the case of juxtamedullary, right, juxtamedullary nephrons, they have the long loop of Henle. So can I say that somewhere they are playing the role here? They are playing the important role here. Yes, please tell me. Can I say so? Can I say so? Right, so mammals can produce a concentrated urine because of this countercurrent mechanism, and that's what I'm discussing right now. So it is a mechanism which will operate in parallel running tubes, right, in which movement of fluid is in opposite direction. So basically, who's going to help here? Your juxtamedullary nephrons, okay, having long loop of Henle. So they play the role in the water conservation, right, in the water conservation and the formation of the concentrated urine. Now see this, see this, look at this part here. This is the descending limb, this is the ascending limb. When you look at these two limbs, the flow of the fluid, the flow of the filtrate is in opposite direction. Yes, madam, it is in opposite direction, right? This flow of the filtrate, how's it? It is in opposite direction. Yes, yes. Look at the blood vessel that is surrounding it. Here also the flow is in opposite direction. Plus, bache, when you look at this part, right? When you look at this part, this is your loop of Henle, this is your vasa recta. Here also, the fluid is, the direction of flow is in opposite direction. So it is the counter current, right? Counter current direction, matlab, it is in opposite direction. One is going like this, another is going like this, right? So when you see, right, so when you see the descending and ascending limb, here also the flow is in opposite direction. When you see ascending and descending limb, the flow is in opposite direction. When you see these two and these two, the flow is in opposite direction, right? And what do you have in the surrounding? What do you have here in the surrounding? This part, what's that? It is nothing but it is the medullary fluid here, right? It is nothing, even it is what? It is the medullary fluid here. Are you getting my point? Are you getting my point? Are you getting my point? Right? Are you getting my point? Something like this. So surrounding me, what do you have? You have the medullary fluid. You have the medullary fluid. See, I'll explain everything. I will explain everything. Right now, I want you people to memorize something because I want to make this topic clear, right? So even I have tried explaining in the context of osmosis and everything. I have seen students struggling, but so this is a very quick wala one shot. So I'll explain it in a very simple way so that you will be able to solve the questions. Otherwise, if we talk about this topic in detail, when we discuss about it, it's very amazing. You'll be amazed to see, right, that how our body works, that our body is actually a wonderful machine. So here they go, descending limb. It's permeable to water. So H2O will come from this side. H2O will come from this side. It is permeable to electrolytes. So obviously your NaCl and all, they will come out from this side. I believe you remember this. I believe you remember this. Yes, I believe that you all remember this part. Right? That this is your water, because from this side water can come out, NaCl come out. Am I right? I hope you all remember. I hope you all remember. Yes, I hope you all remember this. Take that water is coming out from this side, NaCl. So if water is coming out here, it will pass to the medullary fluid. See, that's a very simple thing. Matlab, firstly tell me, this part clear? This part clear? Firstly tell me, this part clear? Yes or no? Is it clear? Yes or no? Is it clear? Yes or no? That from this side water is coming and from that side NaCl is coming. Now, the water from here will enter here in the blood capillary. NaCl from here will enter here. Firstly, remember this. Why is it happening? What is the point? I'll tell you. Again, I'm drawing the same diagram. Right? Again, I'm telling you the same thing. Water is coming out here. NaCl is coming out from this side, right? NaCl is coming out from this side. Take NaCl is coming out from this side. Okay? Okay? Right? So this water that is coming from this side, remember, it will enter into this blood vessel here. The NaCl that is coming from this side will enter here into this side. That is happening. Matlab, jaise, let's say you are throwing out something here, this side is catching it. Like let's say I'm throwing a ball like this, you are chasing it. Like this. Is that clear? Is that clear? First thing is this. Water is coming out here. this side will accept it. NaCl is coming out here, this side will accept it. You know that what's that? This is the descending limb, ascending limb of loop of Henle, ascending limb of vasa recta, descending limb of vasa recta. You know it very well. You know it very well. Let's start it from this side. We just said that ascending limb is playing a minimum role in the reabsorption. That's what I just said. That ascending limb, it is playing the minimum role in the reabsorption. Okay? But it maintains the osmolarity. Look what is happening. What is happening? Blood is flowing from this side, right? Okay, right. Blood is, okay, blood is flowing from this side. Let's say this side. Now, ascending limb is giving out NaCl here. Ascending limb is coming out NaCl here. Try to understand it. Try to understand it. It's the concept of osmosis only. I believe you remember, I believe you know what osmosis is. Osmosis is the movement of substances from there, higher concentration to the lower concentration, from there, higher concentration to lower concentration across semi-permeable membrane. Across semi-permeable membrane. Am I right or wrong? Across semi-permeable membrane. Am I right or wrong? Osmosis is the movement. It is the movement. Movement of what? Because it is osmosis, so we will say the movement of solvent. Right? Because it is the osmosis, then what will we say? Yes, Samir. What will we say? That it is the movement of solvent. It is the movement of water from its higher concentration to the lower concentration across the semi-permeable membrane. Take so osmosis and diffusion. So when you are saying osmosis, you are being specific. You are talking about the movement of solvent. When you are saying diffusion, in general, you are saying movement of substance. Movement of substance it is. Okay. Now what is happening here? Right. The blood, descending limb, blood is going down. Now NaCl. NaCl from ascending limb, it is there in the medullary fluid. Right, ascending limb, it is there in the medullary fluid. It is there in the medullary fluid. Right, I'm explaining you these things in two different ways. It is there in the medullary fluid. This NaCl means it will increase the osmolarity in here. It will increase the solute concentration here. So this DL, it adds the water in it because here it is in the surrounding is more concentrated. So it will enter, it will give some water here and it will reabsorb some NaCl. Because NaCl is more here, less here, it will enter here, plus it will give some water. Now, when you focus on this part, this is the descending limb. We know that it is permeable to water, right? So when it will throw the water in the medullary fluid, when it will throw the water in the medullary fluid, right? The water in the medullary fluid, don't you think this part will become more, this part is kind of become it will be diluted because water is coming out here, water is coming out here, it will be diluted. Water is coming out here, right? So actually what is happening when the water is coming out from this descending limb here, right? Because it is more water here, so that water will enter in the blood vessels, in the vasa recta here, in the capillary here. Because these capillaries, they don't have this thing that I am permeable to this, they are permeable to all. Right, here the simple diffusion is going on. Here the simple diffusion is going on. So from this descending limb, when the water will enter here, when the water will enter here, right, in the medullary fluid, right, that water will enter here in this blood vessel. Matlab, how do I remember? Yeah, see, there is a logic behind. That logic is there in the osmosis and diffusion only, but how can you remember? You can remember simple thing that, okay, this Al was throwing out something, this DL was crossing, DL was like, wow, it is throwing out something, I'll hold it. I will catch it, I will hold it. So NaCl, it will hold NaCl, it will give out water. Now this side, this Al is like, wow, what is here? Acha, H2O. Okay, I want H2O. And this Al will be like, okay, I will just throw out NaCl here. So NaCl is not going anywhere. Right? NaCl is not going anywhere. Just from ascending limb, if NaCl is entering here in the medullary fluid, then through this descending limb, it will enter here in in this part, right? Water will come out, and here, because descending limb is throwing out water, ascending limb is like, okay, fine, here we have more water, I'll take water, I'll give NaCl. Matlab, it's like, see, again I'm saying, the way I'm explaining it is just to simplify the topic so that you can keep it in your mind, right? So that you can keep it in your mind, okay? So that you can keep it in your mind. It is not logical thing I'm explaining, just to put it here. Okay, fine, I'm able to remember the diagram, right? Logically, as I said, osmosis, diffusion, all the things are going on here. Okay, so what am I saying? It's like a monkey, right? Let's say your monkey has picked up your phone, mobile phone. You want your mobile phone back. You want your mobile phone back. You're like, you're showing that food to monkey, banana or some bread or something. So monkey will hold that packet, will throw out your phone, will throw your phone, right? It's something like that. So this side is giving out NaCl. This blood vessel, this descending limb of vasa recta is like, give me the NaCl. I have the water, you take it. Here, this descending limb is throwing out water. This ascending limb, give me the water, give me the water. Take your NaCl back, take your NaCl back. It's something like that. It's something like that. So basically what is happening to this NaCl? It is not going anywhere. If it is entering from this side here, here it is coming out. If it is entering from this side here, here it is coming out. Here it is coming out. So because of that, what is happening? We are able to take more and more water. Right? Because see, if this blood vessel will give the NaCl here, so if NaCl is present here in the medullary fluid, right, then from the descending limb more and more water will come out, because here the constant things will be concentrated, so more and more water will come out. Right? Here we have the H2O here, so NaCl is coming out. That is also one of the ways to remember. Okay, that is also one of the ways to remember. Is that clear? Is that clear? Firstly, is it clear in your mind what is entering, where it is entering? What is entering, how it is entering? First of all, this thing clear? This thing clear? Now, see, we know that, and this is what we discussed. Dekho, ultimately in our body, right, we have to maintain the condition, right? We have to maintain. What we have to do? We have to maintain the things here. We, like, let's say this is one cell. Here in this cell, the solvent is more, solute is less. Solvent is more, solute is less. Here, the solvent is less, solute is more. Solute is more. You know that in our body osmosis will be there. Osmosis will be there. What are we saying? We are saying here they have high solvent, here they have less solute. Less solvent, more solute. It's simple. It's simple. This cell, it is what? It is diluted. This one, it is concentrated. Means water is more here. Here it is less. So you know that the water will move from cell A to B. You know that the water will move from cell A to B. That's the thing. That's the thing. The water will move from A to B. That's the thing. That's the thing. That's how we have to maintain the water. That's how we have to maintain the water. That's how we have to maintain the kind of isotonic concentration. Right? Just again, I'm repeating this part. This is your one cell. This is your another cell. Cell A, cell B. Cell A is having more solute. Cell B is having less solute. Means your cell B is what? Right? Cell B is what? It is having less solute, means more solvent. Okay? So hypotonic. This one is, right? So in comparison to A, this is hypotonic. So you know that water movement will occur. Water movement will occur from B to A, so that there will be a kind of isotonic condition. That's what we know, right? That's what we know. Now what's going to happen? See, these are the cells. Nephron is ultimately what? Nephron is ultimately what? Nephron is ultimately a cell. So you know that in the surrounding, the medullary fluid is there. The medullary fluid is there. We know it very well, right? So we know that when the filtrate, right? The filtrate, because it is coming down. So when you look at this image, see, this is your filtrate, this filtrate. So here if you look at its osmolarity, means solute concentration 300 m osmol per liter. That's how we write 300 m osmol per liter. This is the unit of osmolarity here, 300 m osmol per liter. Look at the filtrate part here. Now when it is coming down, because water is coming down, water will be there in the surrounding. Water will be there in the surrounding. So when the filtrate, it moves down, you know that it will become more concentrated. Just say here, it was 300, then it will become 400, 500, 600, 700, 800. And when it reach deep down here in the medulla, it will be 1200. Its osmolarity is 1200 m osmol per liter. Right? When it is going up, because NaCl is coming out, again, its osmolarity is going down. That's how it is now, right? That's how it is. Now, even in the surrounding, the conditions are same. In the medulla also, in the medullary fluid also, right? This is the condition. So ultimately that filtrate and that interstitial fluid, they'll come to that isotonic stage. They'll come to that isotonic stage. That kind of, right? That kind of exchange is going on. So it's simple. If water, right? If your, yes, this is what? This is your descending limb. This is your ascending limb. So descending limb is giving out the water that will be picked up by your ascending limb, right? So that's why it's Right, that is why what is happening? Its osmolarity is decreasing. Here, it is throwing out NaCl, it will be picked up by this. So that's why its osmolarity increasing. That is how the things are. That is how the things are. Are you getting my point? That is how the things are. Are you getting my point? Firstly, you understand this part. Now, I will read each and everything from the NCERT. Trust me, then the things will be easy for you. Again, I'll show you the more diagrams. Here, dekho. Water is coming out, interstitial fluid in renal medulla. So, ye dekho, these are the sympoters in the thick ascending limb. They causes build-up of Na and Cn in renal medulla. So basically you know that actively or passively, actively or passively, ultimately what's going to happen? The NaCl will be there in the surrounding. So that will do what? That will do what? Maintain the osmolarity there. Countercurrent flow is also there, right? Countercurrent flow is also there, right, bache? Yes or no? Yes or no? See, this condition is clear to you or not? And see, look at this part. This is your descending limb of vasa recta, ascending limb of vasa recta. When it is taking NaCl, it will give water outside. When it is taking water, it is taking NaCl outside. So the way I started, right? Ye dekho, this diagram. See this. This is your descending limb of loop of Henle. This is your ascending limb of vasa recta. It is giving water, it is picking water. It is giving NaCl, it is picking NaCl, then it is giving water. That's how the things are, right? That's how the things are. Now let me read it from NCERT. Trust me, you will get everything. You will get everything. Take, you will get everything. And remember, from the thick part here, NaCl will come to the medullary fluid by active transport, but here it is passively. Chalo, I'll come to this part. I'll read it from NCERT. You tell me if it is not clear to you. If it is not clear to you, right? If it is not clear to you, again read it. See, what are we saying here? I'll go slow here. What are we saying here? We are saying the flow of filtrate into Henle's loop, in the two limbs of Henle's loop, it is in opposite direction. The flow of filtrate in two limbs of Henle's loop, it is in opposite direction. Yes or no? Yes or no? It is in opposite direction. Yes or no? Yes or no? So that is why it is making a countercurrent. That is why it is doing what? That is why it is making a countercurrent. That is why it is doing what, people? It is making a countercurrent. Now, the flow of blood through the two limbs of vasa recta is also in countercurrent pattern. It is also in countercurrent pattern. It is also in countercurrent pattern. Is that clear? Is that clear? Hannah? So the proximity means closeness between the Henle's loop and vasa recta as well as the countercurrent in them. It helps in maintaining and increasing osmolarity towards the inner medullary interstitium. So when you talk about the medulla also, see 300, 400, 500, 600. So when we talk about the deep medullary layers, here they are also the osmolarity, the solute concentration is high, right? It is high, right? So even you know that the Henle's loop and vasa recta, they are also close to each other, right? They are also close to each other. They are also close to each other. Done, right? So that also helps in maintaining the osmolarity. That also helps in increasing the osmolarity from 300 mmol to 1200. So that's also PYQ. Here, right, at the junction of cortex and medulla, you will see 300, but deeply it is 1200. Deeply it is 1200. And all that things, right, all that things are arranged in such a way because we have to take out more and more water from the filtrate. And I'll tell you how is it possible. Okay, I'll tell you how is it possible. So from now, this gradient is mainly caused. How that gradient is mainly caused? This osmolarity. It is caused by NaCl and urea. I just told you the role of NaCl, but not of urea, but I will explain it. Now, you know that NaCl is transported by ascending limb, which is exchanged with the descending limb of vasa recta. Read this line. Read this line. Because I started from this part. Read this line. Read this line, people. I started from this part. Ki NaCl is transported by ascending limb of Henle's loop, which is exchanged with descending limb of vasa recta. If my diagram, if it is clear in your mind, then please tell me. If it is clear in your mind, then please tell me. Done? Yes? Done? Now, NaCl is returned to the interstitium by ascending portion of vasa recta. Right, because we know that near to the descending limb of loop of Henle, we have the ascending limb of vasa recta that will give the NaCl out. So it's like from one side we are picking up the blood vessel, blood capillary is picking up the NaCl, from other side it is giving. Why? Because it has to, that NaCl has to maintain the osmolarity in the medullary fluid. That NaCl is required there, because that NaCl, when it will be in the surrounding, then only from the descending limb, the water will come out in the surrounding, right? That water will be further added into the blood only. That will maintain the balance there in the blood body fluid volume. So, right, so this part clear. Now when you look at this diagram, right, bache, some amount of urea, jase, that's the diagram. Even some amount of urea, bache, some amount of urea, jase here they are showing the arrow, they are, 1 minute. ये देखो, small amount of urea enter the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. So what are they saying? Because we have to maintain the osmolarity here. So if you will see, no doubt NaCl is coming out, but some amount of urea it enters here. It enters here in the filtrate. Then when we will pass from this in the collecting tubule duct also, right? From the collecting duct also, some amount of urea will again pass back to the interstitium. And why it is required? See, urea mane solute there. Then again, because solute is there in the surrounding, so because of that, the water will also come out. That's the whole setup. Okay. So ultimately, it is the diffusion which is going on, right? Diffusion and osmosis which is going on here. Okay. Ultimately, it is like, I can use the word diffusion because osmosis is also a special case of diffusion. So ultimately, it is happening there. So it's simple. From this side, if water is coming, the blood vessel will pick it. From this side, NaCl is coming, the blood vessel will pick it. So when it is picking up the NaCl, it will add some water here. So why? Because that water means more solvent, so solute will enter here. Here, because it is picking up water, so it will enter the solute here, the NaCl here, so that we can take out more and more water from the filtrate. So ultimately, that is the funda, that is the story, that is the whole story here. Okay, ultimately that's the funda, that's the whole story here. Is that clear? Right? So above described transport of substances, it is facilitated by the special arrangement of Henle's loop and vasa recta, and that's why it is the countercurrent mechanism. And this mechanism helps to maintain a concentration gradient. Okay, so look at this part, how the osmolarity will increase from 300 to 1200 mmol when the filtrate will go down. And here again, when the filtrate in the limbs here, you know that because NaCl will go out, then again its osmolarity will decrease. And because we have to absorb more and more water here, that's why from this collecting duct, some amount of urea will also get entered here, because now the solute is there in the surrounding, more and more water will be reabsorbed. That's how it acts. Okay, that is how it acts. Okay, so if it is clear, please tell me. If the osmolarity and everything is clear, please tell me. Quick. If it is clear, then please tell me. Yes, please. Done? So this is about the concentration, the solute concentration here, right? This is about the movement of the things, right, bache? Right, so presence of such gradient helps in easy passage of water from the collecting tubule, and that's why it helps in concentrating the filtrate or the urine, right? That's how the things are, right? That's how the things are. Okay, so ये देखो, descending loop of Henle is water permeable, ascending is not. Ascending actively pumps out the ions. Water goes to where the most stuff is, मतलब where the most solute is, so water will go out there. So that is why NaCl is coming out here. So water will also get out there, right, bache? Then vasa recta will remove the water, मतलब whatever water we are taking out from the descending limb of loop of Henle, vasa recta will remove it, so it doesn't dilute the medullary gradient. So it doesn't dilute the medullary gradient. These are the key facts here. Okay, these are the key facts here. Is that clear? Is that clear? Okay, is that clear? Sure. So presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, therefore concentrating the filtrate. So human kidneys, they can produce urine already 4 times concentrated than the initial filtrate. Imagine, 4 times more concentrated than the initial filtrate. Okay, then the initial filtrate. Understood? Chali, tell me if there is any doubt. So urea and NaCl is responsible for the osmolarity of the interstitial fluid. Okay, osmolarity of the interstitial fluid. So imagine the filtrate, initially it was 300. Its osmolarity was 300 m osmol per liter, right? By the end, it was 1200 m osmol per liter. So obviously, it is 4 times more concentrated. It is 4 times more concentrated. If you think how is it possible, if you again get confused, then again I'm telling you, ye dekho, look at this. The filtrate when it will pass from the ascending limb, again its osmolarity will decrease, but again that filtrate will pass out from the collecting duct. Right, so collecting duct is also adding the urea in the vicinity, right, the urea in the vicinity. So that is why more and more water will be reabsorbed. So when this filtrate will pass through the collecting duct, again its osmolarity will be like 400, 600, 800, 1200. So that's why we are saying that now it is the final urine. Now it is the final urine. And so filtrate, we started with 300 m osmol per liter, urine 1200. So that's why we are saying it is 4 times more concentrated than the filtrate. That's why we are saying that it is 4 times more concentrated than the filtrate. Understood? 4 times more concentrated than the filtrate. Understood? Sure. The diagram, the topics, clear? Please tell me. Yes? Okay, and bache, when you talk about the collecting ducts, you know, even many collecting ducts, they also join. Right, many collecting ducts, they also join. You know, na, renal papilla, if you remember renal papilla. So they also, right, the many collecting ducts, bache, right, those which are in the medullary pyramids, right, they also cross through the medulla in the pyramids, and in the papilla part, na, they converge, they join, and they form a short, thick duct, that is duct of Bellini. That is duct of Bellini. Thick. So duct of Bellini and column of Bertini, these are 2 different things. Duct of Bellini is formed when the collecting ducts, they converge there in the medulla part. Column of Bertini when the cortex is extended into the medulla in between that medullary pyramids. Understood? Right? Right? Sure? Are you sure? Done बच्चे? Okay, just allow me 1 minute. So is it clear? Firstly tell me, is it clear? Chaliye, so let's go. So that's about the osmotic gradient. Fine. So next is the regulation of kidney function, but before that I'll tell you what is that juxtaglomerular apparatus. Take, so that's our last topic. Okay, so maximum I think till 11:30 we'll finish everything. Okay, and that too in detail. So next is JG apparatus, juxtaglomerular apparatus, because now we need to understand the regulation of kidney function. So we will talk about it. Right, we have to discuss the RAS mechanism, very important it is, and there is a separate video as well. Right, if you want to check, there is a separate video as well for this regulation of kidney function. So what we need to understand, JG apparatus, that is juxtaglomerular apparatus. That is juxtaglomerular apparatus. Okay, bache. So here, what are we going to talk about? We will talk about the JG cells, juxtaglomerular cells, also known as the granular cells. And second is macula densa. Now, what is that actually? See, whatever diagrams of the nephron you see, right, whatever diagrams of the nephron that you see, its structure is something like this. Like your DCT, somewhere your DCT, it is, somewhere the things are like this. Somewhere the things are like this. Is that clear? The things are like this. Like this is the arterioles here. Right, they are somewhere in contact with this DCT. I'll show you the picture. But firstly, tell me, is it clear? Because you know that the 1 million, near about 1 million nephrons are there in the kidney. Near about 1 million nephrons are there in the kidney. It's not like that they are having their individual flats. You live there, you live there, you live there. They all are in close proximity. Connective tissue is holding them together. So basically, you will see that these arterioles of the glomerulus, they are in the close contact with the DCT. First of all, tell me, this setup is clear to you? Yes. The setup is clear to you, yes or no? This setup is clear to you, yes or no? Yes or no? Chalo, so let me show you the picture here. Yes, that's the diagram. See this, like it's not that clear, but if you can, the DCT and this part is, see, this is the DCT, the cells of DCT that are touching these arterioles, they are getting modified. We are calling them as macula densa, right? We are calling them as macula densa. Ye dekho. This is your afferent arteriole, the cells which are in contact here. And here in the afferent arteriole, when you look at it carefully, these are the juxtaglomerular cells. They are also known as granular cells. They are also known as granular cells. Right? They are also known as granular cells. Can you see this here? Can you see this here? So the cells of the DCT which are in close proximity. Yes, the cells of the DCT which are here in close proximity. Can you see it here? Right? The cells of the DCT which are here in the close proximity. See this? They are the macula densa. They are the macula densa. This is your juxtaglomerular apparatus. Right, bache? So macula densa cells, granular cells, there are in some books mesengium extraglomerular cells are also given, right? So this is your juxtaglomerular apparatus, right? So if you look at it carefully, ye dekho, these are the granular cells in the arterioles, in the walls of arterioles, right? Firstly, tell me the diagram clear. Firstly, please tell me the diagram clear. Please tell me the diagram clear. Yes or no? Yes or no? Sure? One minute, please. Okay, chaliye, let's write it. Okay, let's write it. So then let's continue. So, bache, JGA is what? It's a special sensitive region. What is it? It's a special sensitive region. It is a special and a sensitive region, right, formed by the cellular modifications in DCT and afferent arterioles, right, bache? Formed by the cellular modifications in DCT and afferent arteriole. And what? And afferent arteriole. So we have two things here, JG cells, which are also known as granular cells. Bache, why do we call them granular cells? Yes. Why do we call them granular cells? Yes, bache. Why? Why? Because they are having some inactive granules, so smooth muscle cells of the, right? So remember, first of all, this is the special sensitive region which is formed by the cellular modification DCT and afferent arteriole. Number two, it helps in maintaining GFR, or basically it helps in increasing the GFR when it is low. So you can say that it is for the regulation of glomerular filtration rate. It is for the regulation of GFR, that is glomerular filtration rate. Is that clear? Is that clear? Right? So what these juxtaglomerular cells are? So the smooth muscles, right? So the smooth muscle cells, what's the point here? The smooth muscle cells of walls of both the arterioles, right? The smooth muscle cells of both the both the arterioles, where it comes in contact with DCT, right? Where it comes in contact with the DCT, they get swollen and have inactive granules, and that is renin. It is RININ. What's the word here? It is RENIN, right? It is not RENNIN. What is that RENNIN? Renin. This is in the, it is in gastric. It is the part of gastric secretions. Gastric secretions. Just in the case of infants, if you remember, right, renin, this is the enzyme that acts on milk protein casein. It is present in inactive form, prorenin, RENNIN, right? So that RENNIN is the part of gastric secretion. So when you are talking about the kidney, it should be RENIN. It should be RENIN. R-E-N-I-N. Is that clear? It should be R-E-N-I-N. So here it should have single N. Just you can remember like this, kidney. Kidney. Kidney is already having one N. That's why its renin is having single N. That's my way to remember. Right? That's my way to remember that kidney is already having one N. So that's why there is a single N there. Right? There is a single N there. Is that clear? There is a single N there. Is that clear? Is that clear? Sure? Are you sure? Take so renin, right? That is the first part. Then it is the macula densa. So they are the cells of DCT which in coming in contact with arterial wall, they become dense, right? They become dense. That's why the word is macula densa. So macula densa is what, bache? Macula densa is what? These are the cells of DCT, the one which are in contact with the one which are in contact with arteriolar wall and they are thick. That is the macula densa, right? That is the macula densa. So this is all about the juxtaglomerular apparatus. Right, this is the point here. So you can see this, it is given in NCERT. Now the diagram part is also clear to you that their kidneys are having a built-in mechanism, right? And due to which they can regulate the glomerular filtration rate. So normally glomerular filtration rate is 125 ml per minute. Normally it is 125 ml per minute or it is 180 liter per day. So how can it be regulated? So they have the kidneys are having the built-in mechanism for the regulation of the glomerular filtration rate. So JGA, juxtaglomerular apparatus, right? That GFR will be maintained with the help of juxtaglomerular apparatus, which is a very sensitive region formed by the cellular modification in your DCT and in afferent arteriole at the location of their contact. So when there is a fall in GFR, it can activate JG cells to release renin, which can stimulate the glomerular blood flow back to the normal. And that's what we need to understand, that how is it possible. Okay, so juxtaglomerular apparatus clear. Juxtaglomerular apparatus clear. So with the help of it, we will also understand, right? With the help of it, we will also understand what? We will also understand the regulation of kidney function. So when there is a fall in GFR, glomerular, see, when the GFR will go down, when there is a low blood pressure, when there is a low body fluid volume. So when there is a low body fluid volume, there will be a low blood pressure, which in return will have low GFR, right? Which in return will have low GFR, which in return will have low GFR. So low GFR will activate that, right? Low GFR will activate that cells to secrete the renin. Okay, so that is all coming under the RAS mechanism. So let's discuss it, right? Let's discuss it. Firstly, let's talk about it. Then I will tell you about the role of ADH, right? Role of ADH. Chalo, I'll make it here only. So when there is a fall in GFR, right? When there is what? Okay. So when there is a fall in GFR, what is this GFR? Glomerular filtration rate, right? And how that fall will be, right? Obviously, when there is a low glomerular blood flow, when there is a low blood pressure, then there will be a fall in GFR. So you can write it down. You can write it down. So this thing is going to stimulate. It is going to stimulate what? It is going to stimulate your JG cells, juxtaglomerular cells. And in response to that, your juxtaglomerular cells will release renin. It will release renin. Now, what that renin will do? But there is angiotensinogen. Angiotensinogen, right? What is it? It is an inactive. Why? Because angiotensinogen, it is not active. It is inactive plasma protein. It is an inactive plasma protein which is synthesized by liver. That's important, right? It is an inactive plasma protein. It is present in plasma, but it is synthesized in liver. What's that? It is an inactive plasma protein which is synthesized by liver. So angiotensinogen. So because renin is released, this renin will act on this angiotensinogen, will convert, will make it active. It will give us angiotensin 1. Right? So it will activate it. So this one is inactive. It is an inactive plasma protein which is synthesized by liver. And what will happen? Angiotensin 1. What will we get? Angiotensin 1. Then this angiotensin 1, it will get converted into angiotensin 2. What are we reading right now, bache? Regulation of kidney function. What do we call it? We call it as RAS mechanism, right? What is this RAS? It is renin, angiotensinogen ya angiotensin aldosterone system. Renin, angiotensinogen, aldosterone system it is. Renin, angiotensinogen, aldosterone system it is, right, bache? So when there is a fall in GFR, glomerular filtration rate, when it decreases, right, it stimulates the JG cells, right? It stimulates the JG cells. What that JG cells will do? They will release the renin. They will release the renin. And that renin will act on inactive plasma protein angiotensinogen and activate it and will form angiotensin. That angiotensin will get converted into angiotensin 2. How? With the help of ACE. It is angiotensin converting enzyme, right? And do you know where will you get it? In the lungs. In the lungs. Do you know that? What's that? It is the angiotensin converting enzyme. It is ACE. What is it, bache? It is angiotensin converting enzyme. It is ACE. Is that clear? Is that clear? So that ACE, right, lungs epithelium is going to form it. You can write down the location. Lungs epithelium is going to form it. And then this ACE, angiotensin 2, it will act on, bache, adrenal cortex, and that adrenal cortex will release aldosterone. That is how it is, right? This is the complete RAS mechanism, right? It will release what? It will act on adrenal cortex and the aldosterone will release. Okay, bache? Okay, bache? Right? So, dekho, firstly, you understand this thing. This angiotensin 2, it will, right? Right, it is also important. Matlab, we have to understand the role of aldosterone as well as the role of this angiotensin 2. So this angiotensin 2, it is a powerful vasoconstrictor. Vasoconstrictor means it constricts the blood vessel, right? It is the powerful vasoconstrictor. It constricts the blood vessel. So if it constricts the blood vessel, see, imagine blood vessels are getting constricted. Don't you think the pressure will increase? Don't you think the pressure will increase? So if it is a powerful vasoconstrictor, then definitely it will increase. Definitely it will increase glomerular blood pressure, and that too will increase glomerular filtration rate, because if glomerular blood pressure will increase, glomerular filtration rate will also increase. And other than that, this angiotensin 2, it helps in the reabsorption of sodium and water from PCT, from proximal convoluted tubule. Okay? So what is it? It's a powerful vasoconstrictor. It increases glomerular blood pressure. It helps in the reabsorption of sodium and water from proximal convoluted tubule. That is the role of angiotensin 2. Plus, it acts on adrenal cortex. So, I believe you all know what is adrenal cortex. See, you know that kidneys, right? Kidneys, they are having one gland, that is your adrenal. Their pair of gland is there, na? Adrenal gland. Adrenal gland, ad means adjacent. Adjacent. Renal means kidney. So it is adjacent to kidney. Cap of kidney it is. It is having this type of shape, triangular shape. So bache, just like kidney, even your adrenal gland, even the ovaries, we see such things there. Outer cortex, inner medulla. Outer cortex, inner medulla. So here, even in the adrenal gland, you see adrenal cortex. What do you see? You see adrenal cortex, and inside, what do you see? You see adrenal medulla. What do you see? You see adrenal medulla. So this adrenal cortex, it is having 3 layers. The trick for that is GFR, G-F-R. That is zona glomerulosa, zona fasciculata, zona reticularis. right? It is having 3 layers. These 3 different layers, they release the different hormones, but we are taking the things and like general things we are considering here. So simply we are saying that the adrenal cortex, it release aldosterone. So always remember, adrenal medulla, it release different hormones, like it will release catecholamines, right? Catecholamines like epinephrine, norepinephrine, but adrenal cortex, it releases the hormones which are made up of steroids, right? So aldosterone is released by this, right? And how that this aldosterone will help? Again, bache, it will increase the reabsorption of sodium and water from the distal part, right, bache, from the distal part. So adrenal cortex, it will release aldosterone, right? And that is the one that is helping in the conditional reabsorption. So again, it helps in the reabsorption of sodium, of sodium and water from distal part, and that will in return increase GFR. That's how it is. That's how it is. That is how the RAS mechanism or juxtaglomerular apparatus, that is how it maintains what? That is how it maintains what? It maintains the glomerular filtration rate. So this topic is the most important one. Okay, this is the most important one. Understood? It is the most important one. Take. So again I'm repeating, fall in GFR, JG cells will release renin, will act on angiotensinogen, which is inactive plasma protein synthesized by liver, right, bache? So this renin will convert this angiotensinogen into angiotensin 1, and this angiotensin 1, it's quite active, bache. So angiotensin 1 converting enzyme released by the lungs epithelium, it will act on released by the lungs, it will act on the angiotensin, it will convert angiotensin 1 into angiotensin 2. So this angiotensin 2 is also a powerful vasoconstrictor. It increases the glomerular blood pressure, in return GFR, and it also helps in the reabsorption of sodium and water from proximal part, from PCT, right? And this angiotensin 2, it also acts on aldosterone, in return it also acts on adrenal cortex, which will release the aldosterone, and that aldosterone will also help in reabsorption of sodium and water from the distal part. Is that clear? So this is the RAS mechanism. You can see the arrows here, right? So surface of pulmonary and renal endothelium will give you the ACE. Surface of pulmonary, that is lungs, and renal endothelium will give you the ACE, which will form angiotensin 2, right, bache? So I told you how will it work. It will also act on adrenal cortex. It also a vasoconstrictor, right? And somewhere it also, as per this chart, it is having a, it also somewhat stimulates the ADS secretion, right, bache? So again, aldosterone, it helps in tubular sodium and chloride ion reabsorption plus potassium excretion. So I just told you that it helps in the sodium reabsorption. Sodium reabsorption plus potassium excretion is also the important point, right? So the water retention will occur, right? So sodium will be reabsorbed, potassium will be excreted out. Sodium will be reabsorbed, potassium will be excreted out. Sodium reabsorption, potassium excretion. Okay, potassium excretion. That's how the water will be retained. Okay. So angiotensin 2 also stimulate the ADS secretion. Done? Sure? And the role of aldosterone and everything clear? So do not forget sodium reabsorption, potassium excretion. So this is also one of, so ACE acts on angiotensin 1 to form angiotensin 2, which is vasoconstrictor, plus it is also acting on adrenal cortex. So there are different flowcharts that I have uploaded here, right? I'll definitely share, right? I will definitely share the PDF in the Telegram group. Take. So now let's discuss about the ADH. Dekho, your hypothalamus, which is the super master gland. What is thalamus? What is it? It is the super master gland. What is it? It is the super master gland. You know that it acts on pituitary. Pituitary, which is also known as hypophysis, right? And pituitary is the master gland. So you have two things here. You have adenohypophysis. You have two things here. You have adenohypophysis and you have neurohypophysis. neurohypophysis. So when you talk about the adenohypophysis, it will include your pars distalis, that is the anterior lobe of pituitary, and it will also contain pars intermedia, that is the intermediate lobe of pituitary. Then this neurohypophysis, you are talking about the posterior lobe of pituitary here, that is pars nervosa. So you know that this posterior lobe of pituitary is under the direct neural regulation of hypothalamus. It is under the direct neural regulation of hypothalamus. So this pars nervosa, bache, this neurohypophysis and this pars nervosa, because hypothalamus has synthesized that hormones are stored there in neurohypophysis. So when there will be a signal, because posterior lobe of pituitary, it will release two hormones, ADH, anti-diuretic hormone, also known as vasopressin, and oxytocin. So here we will deal with ADH, that is anti-diuretic hormone. That is anti-diuretic hormone. So anti-diuretic. Diuretic means diuresis. Diuresis means excess of urination. Diuresis means excess of urination. So if it is excess, so if it is ADH, right, that is anti-diuretic hormone, means it prevents excess of urination, right? It prevents excess of urination. So this ADH is also known as vasopressin, right? Because it is also having a vasoconstrictor role. So it is also known as vasopressin, right? It is also known as vasopressin. Is it right? So osmoreceptors, right, that can sense these kind of changes in our body, they are in the body, they are in the hypothalamus, they'll be activated, right? Osmoreceptors, right, which can, just receptors, osmoreceptors, they can sense the acidic base balance, yeah, salt and ion concentration in body, right? They can do that. Done. So osmoreceptors in the body, they are activated by changes in blood volume, body fluid volume, and ionic concentration, right? They are what? They are activated by changes in blood volume, body fluid volume, and the ionic concentration, okay, and the ionic concentration. So when there is excessive loss of fluid, when in our body there is less fluid, so ADH, the osmoreceptors, right, this can activate these receptors, that receptors will stimulate hypothalamus. So hypothalamus will release ADH. How? Hypothalamus will tell it to posterior lobe of pituitary ki please release ADH, also known as vasopressin, from neurohypophysis. So ADH helps in the water reabsorption from the later parts of the tubule. Later part, your distal tubule, your collecting duct, right? And that is why it prevents diuresis, means excess of urination. It will prevent excess of urination. And when there is an increase in body fluid volume, then again these receptors, they'll switch off or suppress the ADH secretion. That's how it works. Plus, I told you this ADH is also having the constrictory effect on blood vessel. See, the word is vasopressin, will press blood vessel, means having the constrictory effect. That's how you can remember, right? That's how you can remember. Okay, students, that is how you can remember. So this is all about the regulation of kidney function. I told you the role of ADH. I told you how it acts. I told you how angiotensin 2 will also help in the release of this ADH. So this is how everything is related. So just let's say when we drink a lot of coffee, tea, ya fir even if somebody, right, is alcoholic, in their case, they urinate again and again, right? When we drink such kind of beverages, we urinate again and again. Why? Because already in body fluid, the volume level is, body fluid volume is good, right? There is no need to reabsorb the water. So excess of urination is there. So you can see that, that these kind of beverages, they are responsible for the diuresis. Right. So when right, there is a water scarcity, then obviously, and when we drink such kind of things, then there is low ADH in our body, right? When we don't drink that, ADH level is high, so it will prevent the diuresis. That's how the things are related. That's it. So these are the important topics. Rest everything is very, very, very simple, right, bache? That's everything is very, very, very simple. It's simple. Ye dekho, that urine formed by the nephron is carried to urinary bladder. It is stored till a voluntary signal is given by CNS. So CNS will give a voluntary signal, right? So there is the stretching of the receptors. There is the stretching of urinary bladder. That will give a signal to the brain, right? When the urinary bladder, it gets filled with urine, the stretching of urinary bladder, right? The stretch receptors are present there. They will send the signal to central nervous system. So you know that signal will go by a sensory message, sensory neuron, and the motor neurons will, right, motor neurons will tell us what to do. Okay, what to do. So that is how it is. So ye dekho, look at the urinary bladder, the sympathetic, parasympathetic nerve fibers, they are innervating here. Here you have the sphincters, right? And I told you external sphincter, it is what? It is voluntary. Internal sphincter, it is involuntary, means, right? Right, it is involuntary, I told you already. So when you talk about the passing out of urine, the urination, for that we also use the word micturition. Micturition, right, bache? So when the signal will go to the CNS, the motor message will initiate the contraction of smooth muscles. So when there is the contraction of smooth muscle of bladder, because this is involuntary, right, bache? So contraction of smooth muscles of bladder, there will be the relaxation of sphincters. In the starting itself, we have discussed that sphincters, when they are contracted, your urinary bladder is closed. When they are relaxed, the urinary bladder is open. The smooth muscles, they are contracting, they will pass out the urine. And we discussed the detrusor muscle, which is there in the urinary bladder, that is having that expelling out action. Okay. Okay, so micturition is a neural mechanism. Right, bache? So all that things we have already discussed. I told you when there is a malfunctioning kidney, the glucose, if it is present in urine, the condition is glycosuria, and ketone bodies, water soluble bodies, they form when, you know, there is a, like when we don't eat food, when there is a long prolonged period of starvation. Do read about the ketone bodies. So ketonuria, these two things present in the urine, they indicates diabetes mellitus, right? Diabetes mellitus, understood? Diabetes mellitus, understood? Okay. Done. So then bache, other organs, you know that carbon dioxide approximately 200 ml per minute, right? It will be removed, and the significant quantities of water, right? Lungs will remove it, right? Lungs will remove it. Who will remove it? Lungs will remove it. Liver, which is the largest gland, it secretes bile containing bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamin and drugs. So it also removes the waste. Right, bache? So along with waste, these things will also pass. That's why, because of these pigments, the color of, right, our excreta is having that particular color accordingly. The sweat glands, the sweat, the sebaceous, the oil glands, they also eliminate certain substances through their secretion. So sweat, bache, it is a watery fluid, the having NaCl, small amounts of urea, lactic acid. Okay, even it is having the lactic acid. Baki bache, primary function is sweat to facilitate the cooling, because you know after evaporation there is the cooling. So that's it. Sebaceous gland, they also release certain substances like some sterols, hydrocarbon, wax through sebum. So saliva is also having, you know, some amount of waste. Baki disorders, bache, see, when kidneys are not functioning properly, if urea is accumulated in the body, that is the uremia. And if this condition, if urea will accumulate in our blood, that can just result in the kidney failure. And if there is a kidney failure, then bache, either the artificial kidney or kidney transplantation is the solution, right? So urea can be removed by a process called hemodialysis. So whenever you talk about the hemodialysis, na bache, the diffusion, right? It depends upon the diffusion. Okay, it depends upon what? It depends upon what? It depends upon the diffusion. Just say what's going to happen here, right? Right, from the veins. Just for the hemodialysis, what has to be done? Because there is a proper setup. There will be a cellophane tubes. Again, they are semi-permeable. They'll be covered with the dialyzing fluid. The dialyzing fluid is having the same composition just like plasma of our body. The only point is, in that dialyzing fluid, the waste is not there. Right? The waste is not there. So what is happening here? That during the process of hemodialysis, right, the blood drained from a convenient artery. Right? The blood will drained from a convenient artery. It will be taken from a convenient artery, but it will be returned with the help of a vein. So from the convenient artery, it is pumped into a dialyzing unit, right? That dialyzing unit is actually the artificial kidney. So, okay, picture is not there, but you can say that that dialyzing fluid will have some, you know, some tubes that is covered with the dialyzing fluid. It's simple, bache, this dialyzing fluid is having same composition just like plasma. So whenever that blood will come out, pass through this. So obviously as per the concentration gradient, because here you will have more waste, here you will have the waste, the nitrogenous waste in the surrounding, it is not there. So it will come out there in the surrounding. So that's the principle here, right? That's the principle here. So blood will drain from a convenient artery, bache, right? It will be pumped into a dialyzing unit. But before that, we have to add anti-blood coagulant, because when we are taking out the blood from the body, we do not want that blood to get clotted, because in the body heparin is there, but outside the body it is not there. So this is something that we need to add, right? This is something that we need to add. Okay, so anti-blood coagulant like heparin will be added. The unit will contain a cellophane tube which is surrounded by a dialyzing fluid. So this dialyzing fluid, that's a PYQ. It's the same composition as that of plasma, but it doesn't contain the nitrogenous phase. So this porous cellophane membrane of the tube, it allows the passage of molecule based on concentration gradient, right? So as the nitrogenous waste, it is not there in dialyzing fluid, the substance will freely move out, so it will clear the blood. Okay, so this cleared blood through the vein, again it will be entered in the body, but after adding anti-heparin to this. Okay, so when heparin is added to the blood now, then it is cooled to 4 degrees Celsius. Okay, so then again when we are returning the blood back to the body, then we have to bring it to the normal body temperature, then the anti-heparin is added. Okay, so it is depends upon the principle of diffusion of small solute molecules through the semi-permeable membrane. That's it. That's it. Okay, that's it. Fine. Fine. So baki kidney transplantation for the renal failure. Okay, so transplantation will be done. Baki renal calculi, you are talking about the stones here, right? When we are saying renal calculi, right, you are talking about the stone, crystallized salts, right? They are the oxalates and they are crystallized salts. Okay, so disorders are very important, bache. So renal calculi is also known as urolithiasis, right? For that, there is one more word, urolithiasis. Okay, then this is the glomerulonephritis, which is also known as Bright's disease. It is also known as Bright's disease. It is the inflammation of glomeruli, and that is because of bache, any reason can be there. There can be any infection or injury in the basement membrane that can result in the renal failure as well. Okay. Okay, so bacterial infection can cause that. Protein and RBC will start passing into the filtrate because the pores, they will get widened. So that's how it is. Baki bache, when there is a hyposecretion of ADH, then it can result in diabetes insipidus. Okay, that is one disease. It is not mentioned here, but still you can write it. Still you can write it, that diabetes insipidus. Insipidus. Okay. Diabetes mellitus, when urine is having glycosuria and the ketonuria. So diabetes insipidus, right, when there is the hyposecretion of ADH. So less secretion of ADH, right? Less secretion of ADH means more diuresis, right? Again and again that person will urinate. So because of that, body fluid volume will also goes down. That person will be having polydipsia, under a constant thirst. Okay, so do read about it. So this is all about this chapter that again we have discussed right properly. We have covered all the possible points here, bache. You solve the PYQs, you will be able to answer each and every question, right? Trust me, after the class, give it a try. Give it a try. And do not forget about the biggest event, bache, that is your Aram 2025. So what you have to do, yes, do like this video. Okay, just a minute. Yes, so it will be live in 11 days, that is on 1st March 7 PM. So just go to our channel, right? Just go to our channel, so you will see it there. Fine, you will see it there. Done, bache. And in the description box of this particular video, there is a link for our Dream Crash Course batch. You can click on that link. You can be the part of the batch as well. So please revise properly. And if you like the session, do let me know in the comment section. And which chapter you want me to start next. I am planning to start chemical coordination and integration. Let's finish the human physiology, then plant, then genetics, reproduction unit. What say? What say? So guys, if you like the session, do let me know in the comment section. I'll share the notes in our official Telegram group. So stay tuned, take care, thank you. And yes, no doubt you have to fill the NEET application form. But yes, recently I have posted one video on Unacademy NEET English regarding the essential forms, the other important forms that you all should fill. So make sure you check it, make sure you fill it. Thank you so much.